is increased. And here, through the collaboration that was also with researchers from the Catholic University and Daniela Watzlet, who was also looking at some of the strains of this pathogen, and with researchers with the part of the ring who had access to these strains who were evaluating this, we started to sequence a collection of around 30 to 40 genomes, 50 genomes of Streptococcus pyogenes with Nicolas Canales, who is bioinformatics, and he started to do the comparative genomes. And what can we see? We can see in the tree most of the strains. The red that you can see is mostly from Chile. The ones I do not have where I can point it to you, but you can see that it is close to the ones from the United States and from England in the bottom part of the tree. We also noticed, and recently from Argentina, there is a work that was in Argentina that published the strains, and there is this particular strain that is kind of to the right. We also have some from Argentina that are very close to Argentina. And we do not have the data. We need the depth of data to be able to state that the strain of Chile came from Argentina or that it went from here towards Argentina or from Argentina to Chile. And this is something that is very important because there are few genomes as a region that allows us to carry out this kind of study. So, as we have more information and more public information that is available, we can start improving the resolution of this kind of analysis and understanding how it has moved. A little bit of like what we were showing before with SARS-CoV-2, like the geographic dynamics of the mobility of these organisms and how it has been changing over time. And in particular, connecting it with what we mentioned previously of the M1 UK with what we are seeing here. So, and also from what we saw in the SARS-CoV. And from this collection of strains, we can see that around 25% of these strains were M1 UK. We were not looking at a replacement as we did, for example, what was happening in England or other countries in Europe where it was dominating. But it is starting to show up. And then this gives us the idea of this surveillance. We have to be checking to see if it is appearing, if it is cohabitating with other variants. But what we are finding here is that some of these were from this variant M1 UK, and they are similar mostly to those isolated in England, but also they are also similar to those because those are the closest ones. Maybe they could also be very similar to isolates that are in other countries of Latin America. However, we do not have the genomic problem information to be able to place this in context. So this is a view of how um, through these genome tools of bioinformatics, we can start asking the genome questions that require to generate data and asking questions regarding surveillance and how this organism is changing over time and how it is moving in a geographic way. And to be able to end going to another level of the analysis, I started from a virus and from more of those genomes that have been isolated. The last work, and this is something recent in the data, it started a few years ago, but now the last 3 months we have had the strong part having to do with data, which has to do with what happens with these pathogens if they're resistant to antibiotics, but in metagenomics and antibiotic resistance, and how these patterns of resistance change over time in populations and individuals. And this starts for the collaboration within this ring with Dr. Rafael Arauz, co-director of the ring, where he had a cohort that were carrying out the colonization of resistant microorganisms, and these are patients that were hospitalized. And every 7 days, when they first entered, they would have a swab, an anal one, a rectal swab, and that way they would keep the sample so that we could reuse it to be able to have a DNA. And this was done every 7 days until 9 visits, supposing that they would continue in the study. Not all of them have all of the visits. that were required. But in these samples, there are studies that are being carried out in the cultures. For example, what microorganisms are we seeing? What resistance do they have? How is this resistance changing over time? But then we have the opportunity that we also have genetic material that has been stored so that we can see not only the microorganism that was stored, but also what happens with the whole community and the people of this study.
This is for you to see what happens with the numbers, to have an idea of what happens with the numbers. This is the amount of samples we took, and with several visits, we have the samples of the first visits, and for some reason, they didn't continue with the study. They were discharged, they didn't want to continue, and the participation changed in time. And for some, these they had microorganisms that were resistant and they were colonized, and some there was no colonization. But this shows us how it changes in time. A lot of these people that had colonization, there were colon. I'm sorry, that had no colonization, uh, didn't have anything in the beginning, but then in time started to acquire the bacteria that we were focusing on in this study, those resistance bacteria with cephalosporina as well. And what we saw here is the information that we obtained from the cultures of E. coli, Streptococcus complex, with these resistance that this study was focused on. But now we had the opportunity of studying the entire community. And through this, Francisco, who was part of the workers of the lab, and is some of the few things that we've done in the lab because we do much more in the computer. And this was a working collaboration. We worked with a startup called Draker is the name, and they helped us with the entire process. They basically helped. Lended us their lab and their extraction tools and and we were able to do the sampling with their tools and not um, by hand, and we were able to do it with their help. We took 798 cards, and not all of them were successful. We were able to sequence successfully about 510, I'm sorry, from the 788. And for those who have worked with genomic data, what we have here is about 97,433 million of reads, which is about 14 terabytes of the entire data set. So this is data that we received in February, and due to logistics, it took 2 weeks to download them. And because the conditions were slow, we had to start downloading it from home. It was a whole logistic issue. We had to make space for the data, and we were optimistic that this was going to work, but we never thought we were going to have so much data. And that's what I was just mentioning, the development of tools. And this is what we were getting ready for. We know that we will have the approach of the resistance to antibiotics, population changes, so let's get ready and be prepared for this. And during that time, as we were waiting to receive all of the equipment, Francisco received a pipeline that was called Backbuster, and it's focused on the analysis of the metagenomic data by characterizing the species that we found with a focus on antibiotic resistance as well as predictions of resistant genes and specific mutations that can be found and SNPs associated to resistance that can be found inside the sequence, and with a perspective of considering the data and the microorganisms that are present, not just thinking about, okay, we found these bacteria, these genes, and we started thinking about how to rebuild the genomes of the microorganisms, the assembled genomes. So we have a collection of readings and genomes, readings that come from this community, and with that we can rebuild the organisms that are present, because that's what this allows us to do. We can later compare those that we found with the ones that are isolated, and we can go and take those isolated that we took from the same people and sequencing and check if we're seeing the same thing. This is a pipeline that's also available, and the paper is under review at the moment, but it's also available as a preprint if you want to take a look at it. So what are we started seeing from there? We have a lot of data, and we're just getting started with what we are observing. And something that I asked Francisco, and was very useful for today. I said, let's focus on some individuals that had samples throughout their visits. And there's one person that completed all visits. We have samples of each visit, and the person started without a colonization, and from visit number 4 onwards, we started to see colonizations. So the first samples, no colonization, and from visit 6 on, we saw colonization. And what happens when we start looking at this from the data, and by sequencing this data? And for those who have seen microbiome studies, this is the diversity that it has. This is how it looks. We're able to see then that this person in the beginning, the community has Bacteroidota firmicutes, which is very common in humans. From visit 3 on, we see that there's something strange because there is an actinobacteria, and this is something that I'll share with you later, and the reason why it's very interesting. Then we see that from visit 6 on, it stabilizes. And when you look at it in detail, when you look at the species, I want to highlight visit number 3, because in visit number 3, we see this gray bar, a species that is not present in the other visits, and it's not very big. And it was interesting. Francisco started looking at it from up close, and we realized the following. On visit 3, we had less DNA, we had less readings that had passed the filters, but for those that had a 60% were dominated by Microbacteria tuberculosis. And we're still looking at it from the point of view, okay, this is the data, this is what we have. We still need to do the connection from the clinical aspect to check and if the person was hospitalized because they had Mycobacterium tuberculosis. And the sample that we observed when we analyzed the data, this was the sample that had the most human DNA. So our data was more contaminated with the DNA of the person, something that if you're from the informatic area, one of the things that we had to assess was that if the person had tuberculosis, is ingesting more of the mucus, and that's why that affects the data that we are observing. And that's the explanation we were giving that. And we see this that there was a Mycobacterium tuberculosis, and we still need to do a link with the clinical aspects to understand the reason of why this happened. And this is the main question. What happens with antimicrobial resistance? We can see that the resistance, it's low, but then it increases from visit number 4. And we had no predictions, we found no genes, but that's because the depth of the data wasn't as deep. And then we found that from visit 4 on and visit number 5, there is a gen uh, FX, CFX. It's a lot. You see much more from visit number 5 on, and with the idea of relating the data, the culture's genomic data, to try to understand if we're finding the same thing in the different kinds of population, we can agree that we are capable of detecting with the information that we have with the sequencing and of the Enterobacter colicle and Escherichia coli. Um, and just to give you this idea, and with this I conclude, but with the 9 samples that we took from this person, we were able to recover about 103 genome or different kinds of bacteria species. We just want these samples. So what we want to do at the population level, with all of the data that we will have, we want to try to rebuild a lot of these species and these abundant organisms, and try to understand how is this relates to the cultures, and whether what we culture, is it the same thing that we are finding in the samples, do they have the same resistance. And going back to what I was showing you earlier, and if we do a tree, does that tree matches to what we're seeing in the sequencing and for a specific person, and when isolating the strain, and how the individuals colonize in time and develop a resistance in time. And this helps us answer a lot of questions, and we can do so with the predictive modeling and all of those areas will help us figure that out. And this is the comparison I was just explaining. And with this, I conclude because I don't have much time. I would like to thank the research team that helps me with these ideas and the craziness that we're in. And this is all financed by a need and the gene to this, because we look at the genomic data, but with this, we are able to put this in context so that it is relevant in clinical aspects and health aspects. And CDC also is part of our project. Thank you very much. Thank you, Juan. The floor is open for questions. Hi, Juan. What you it's very interesting what you are showing. The study of colonization and how it behaves with different pathogens is something that creates information for risk management within surveillance of microbiology. And it is interesting to compare this because we're doing something very similar in the ebiogenes, and we have several elements that we have sequenced, and we go to a lot of clinics and we look at the phenomenon of co-detection and the fatal outcomes if or whether the patient recovered. And we're looking at that, and we are trying to find the best informatic model that adapts to our reality and our informatic capabilities. And of course, um, to have a sampling system, because it's impossible to sequence everything. And something that I discussed with Rafael Arauz is that it is important to know this information and try to maybe have meetings so that we can talk about what we are doing, because we are not specifically focusing colonization, we focus on the confirmation of cases. And it could be interesting to understand what you are doing as our counterpart. Yes, we should talk about this. I would be happy to discuss it. And from me, the question is more from the metagenomics side, and that was the line I would work with. And with the collaboration we've done, we were told, okay, this is study, and what we were going to do was just characterizing the changes in the community. But at some point, we said no. When we started thinking about this 5 years ago, the cost had decreased, and we had we could finance some of those projects, and we said, okay, let's try to do this. And the worst case scenario was, okay, maybe we're not going to see a lot of colonization, but from the knowledge that I have to the date, this is the biggest microbiome characterization that we will have humans. And with that, we can do some other things in the future and take that to the health. Um, uh, and try to understand the genetic factors, risks, etc. For the modeling for the future and everything that has to do with the control of friction. I think what you're showing is very interesting, and it's something that complements what we are doing as a surveillance institute. So it's very interesting. Thank you. We have time for a question back there. Dr. Ramirez? Thank you. First, congratulations. Great job. And I think that it forces us to look at the pandemic differently, among other things. And from the point of view that we work with, which is virus and SARS-CoV-2, I have two questions. One, sequencing, and what you were presenting, which is an entire genome or SPI. And that's the first question. And the second question, when you do sequencing for SARS-CoV-2, do you do this sequencing homogeneously or do you have populations? And I'm asking this question that seems shallow, but I see that there are several subvariants are coming up, and although it seems that we're all vaccinated today, and therefore there's a selective pressure against these viruses that's very different than what we had in 2020. Really good questions because specifically that second question, this is something that I have been thinking for a while now. The first question, the trees are entire genomes, genomes that come from GSI, from public databases, and in the case of the Chilean genomes that have come from ISP and different reference lab, um, these are all complete, not just the spike, but the quality control had to be done without contamination in order to use them. Regarding that second question, this is something that I agree, and I have been thinking about this since the beginning of the pandemic, because when we started sequencing, in the case of this virus, we're doing the sequencing, and with that, we're agreeing on something, and with that, we are losing that variation of population because that's what we're seeing in the genome. And I haven't been able to convince anybody in the lab, and I haven't been able to look at it either. And one way of doing it is to have the consensus. We have the crude data of what we do in each platform, and this also means that you cannot take this public data, or you take the public data that people have uploaded, or in addition to the sequencing public data, or that's because you are sequencing that and you have access to that raw data and you saved it. And and sometimes they use space, and I'm always saving stuff in real life and also in terms of data. But this is something you could do. You can start looking on that agreement, compare the sequencing data, and look at populations, look at where are variants, populations, and where in the genome the variants are showing up. And why do I think about this? And we can talk about this because my doctor's thesis, which was in microenvironment, we study populations in bacteria with those same questions. So it's very interesting. And thank you very much for that question. Thank you, Juan. And we're going to ask our head of the department to come to the stage and give you an award for the participation. Thank you. Let's continue. The topic of the next conference is called Biofilms of Aeruginosa pseudomonas, multiple strategies to persist in the environment. In charge of Gerardo Cárcamo from the Catholic University of Chile. He is a biochemist and works in biology in the University of Concepcion, doctor in microbiology of the University of Zurich, Switzerland, and he has had a worked in Massachusetts, MIT, in a postdoc. He has been a professor in the Catholic University of Chile, shared position between the Faculty of Biologic Sciences and the Medicine department. His research is focused on the study of bacterial interaction, including the formation of biofilms, cellular communication, and contact competence, and the contact of in the context of interaction with the environment. Dr. Carcamo, the floor is yours.
Can you hear me okay? Well, I would like to thank the invitation and the opportunity to be able to share the results with you. The truth is that I still feel like I have just arrived in the university, so I have not had the opportunity of sharing the amounts nor the quality of the results that we have seen. I would like to take advantage of this moment to be able to, based on the different stories that I have worked on during my scientific career, to be able to have found a line or an objective or to be able to understand a little bit of the microbes that applies perfectly in the vision of a comprehensive health that we are called to work with today. It is not necessary to start my lecture like this one, but we have to reach audiences that are not necessarily microbiological or that ask you, why are you working with these bugs? But to begin, I would like to show this picture that we see here that is considered one of the best representation, graphic representation of our planet, and it was by the Apollo 17th. This picture was taken in 1972. And I consider that this cartoon that was carried out by the microbial health earth of AS, created by ASM, is just as good because it shows that in our planet the microbes are the most successful form of life. They are the organisms that are in all of the environments that we know. And if we can understand this main evidence, we will be able to understand that if we can understand the microbial behavior is key to be able to understand the problems that we are dealing with as a civilization, and many times be able to find in the strategies in the microbes, we can have alternative solutions for the problems that we are dealing with. For example, the different sources of energy, different alternatives for food, and also the challenges that climate change includes. In this sense, microbes in general, as individual microbes or as communities, can be used to be able to understand this phenomena and find solutions from different alternatives. To simplify this, microbes can be very good biomarkers that allow us to understand how environments change. For example, what factors are involved or what um, the microbes have gone through different samples where it characterizes the communities that are there and associates the external changes in these communities. They can be used to inspire solutions. For example, in the particular case of Chile, there is many strategies that have used the microbes to adapt, for example, to environments with more salt, less exposition of water, and now they use it to keep the water closer to the roots in some different crops. And another consequence of climate change is that we have new challenges and that are very multiple, and very close to that is what we are going to talk about today. I consider myself mainly a laboratory scientist, and we start looking at a lot of different things, and we work with complex conditions. So we have a lot of different conditions, and we are all day in this lab, and this has allowed us to reach an approach to the behavior of the microbes, starting with the classical interactions that has dominated the study of microbiology, at least in the last 50 years, that once we have a microbe in the laboratory, the idea is to ask, how does it grow? If it does not grow, with antibiotics, if it grows, if not, it's resistant. If it doesn't grow with carbon source, then that means it doesn't eat this and that. That's what explains why it's in certain places. This allows us to study different changes and attributes as they change, and things that happen in different phases, for example. However, many of the behaviors in real situations, whether the environment or others, happen in microbes that are not isolated. They are in interaction with other microbes, and a lot of the results of the structure and the function of a community, in other words, the name of the different microbes that are there and what the communities do all together, depends on multiple interactions. These interactions, like for example, to prepare the ground so that then another microbe can colonize certain surfaces. Interactions, for example, that I eat all the food so I do not allow another microbe that is slower to be able to eat, or I produce different residues that ends up being a source for others. All of this interaction, to be able to identify it and to be able to study it in a systematic way, means that we have to study complementary approaches that are much more simple. Because when we increase the level of complexity to be able to work with a microbe, it can be complex, and we have two or three together, then it's even much more complex. And the cultures that allow us to do that is very limited. And if we also want to be able to study how they behave amongst themselves, then we have to generate the methodology that allows us to do so. It is a challenge, and most people work only with one. Now, an aspect that we have to consider is that nature microbes come in communities where they interact with each other. And a way of interaction, one of the physical interactions, is to create structures with bacteria, which is called biofilms. In the last 3, 4 years, there has been a redefinition of some of the concepts that we will look at in a little bit. But the main thing is that when we have had estimations regarding or considering all of the biomass, for example, we have considered that on our planet we have around 1.2 times 10 to the 30th bacterial and archaea percent, and 40 to 80% resides in biofilms. One of the problems that we saw in the field is that for a long time they considered biofilms as something unique. A lot of us read different papers where they would talk about, for example, 5 stages of the formation of biofilms. There was a bacteria that was swimming, it got stuck irreversibly, etc., etc. And as science, these are book models that are very attractive and very nice because we can build on them. But the truth is that it was, the engineers would say, they were overcomplexing, making overcomplex, because there are some bacteria that don't swim. Not all the interaction and microbiomes are between a liquid and an inert surface, etc. So the new convention, based on a lot of literature, suggests that the formation of biofilm is the formation of aggregation on the surface, aggregated amongst themselves, aggregation of particles, of particulate material in the air of Santiago, for example, or in the water column. And the key point is that the same cells, the identical cells, are being a part of a biofilm. They behave different than what they do when they are not part of a biofilm. And the challenge that we have is that if the majority of the questions that we ask about bacteria are done using mechanisms like these, where we make them grow in with a lot of nutrients, with a lot of concentration of oxygen, we may not be able to respond to some specific questions. And one of the challenges is to be able to generate mechanisms to understand these communities better. But to be able to answer these questions, we have to have study models that are relevant. And throughout my career, this happened because a student is assigned with, you assign the microbe that you are dealing with. But in my master's and doctorate and postdoctorate, I have worked with different pseudomonas. These are some that are pathogenic and some that are not. And if there is something that is common amongst them, is that they are in a lot of different environments, and most of their success is based on the biofilms that are formed. The biofilm is not necessarily something bad, but in what sense? The gender of the pseudomona in general is a good model to be able to ask questions like, for example, what are the consequences of bacteria that have biofilms in the environment and how they can affect as pathogens and other things. For example, we can assume without making a mistake that the same strategy, microbial strategy used to be able to adhere to a microplastic, is going to be the same strategy that is going to be used to be able to adhere to a catheter in a patient. The same strategy to survive thermal variation on a plant is the same one that they use to be able to live in a corner of a hospital that is not so well cleaned. So to be able to understand that mechanism, it is going to take us to be able to understand the transmission of diseases, process and other things like that. So the first study case, and I'm going to try to go quick. This is like a sample of the kind of experiment that we can carry out using as a model. This is, Pseudomonas putida is a bacteria that promotes the growth of plants. And when I started working on this, we knew that communication process in a cellular way was generating biofilms. And that was very relevant. So we carried out a work of basic science where we described molecular uh, part of behind this association, but it allowed us to establish the methodology that allowed us to discover that we never thought we would be able to discover. And this is a summary of that. And what we can see here in green, Pseudomonas putida SOF, if we see this individually and we combine it with different kinds of bacteria, many different bacteria that have not been dyed that under the microscope are going to be seen transparently. And if we put in the middle a fluorescent that will only go into the cells that die or who have their membrane damaged, then we can see. What we consistently saw is that if you incubate them together, and then literally you just look at what happens, this was me hours just looking to see what happened, we can see that after a time, what we see is dead cells, but not in any area of the sample. If we look at it in detail, the first dead cells are those who in the first picture look like they have in direct contact with green cells, no other one. If we wait a little bit longer, we can consistently see the same, that the cells that die are those who are in contact with the green cells. The name of this competitive cells is more complicated. We can summarize as many different pseudomonas that are associated to the growth of plants, but we can also see on the right the different phytopathogens. And what we see here is if we can mark a bacteria with a fluorescent plant, this time we used a red protein, so we don't have to look at it through a microscope. We can see the fluorescence of the colony, and we combine it with our strain that is being an antagonist. And what we can see is that when we have monocrops of the red strain, they become fluorescent, and when we combine them, they lose the fluorescence. That means that the death or then we can see this colony, and in summary, we could see that our Suriname putida ISOF had different gram-negative reactions. They had contact-dependent antagonism against wide range of gram-negative bacteria. So to be able to, now we started a mutant library, and this is in each system, in that each gene of the genome makes for this agent not to work more. And you can ask questions like, are we going to find a mutant that is not capable of killing the competitor? And based on the fluorescence, we isolated thousands of mutants. We made them compete between a red bacteria, and if that bacteria continued being red, that means that it wasn't capable of killing it. So what we found was the genome, sequencing the genome and identifying where the insertions were. We found that all of the insertions that we could identify were within a genome island that identified a secretion type 4. There's a lot of different secretions. Just follow me with this. There was a cluster of genes, and in general in the bacteria, and the biological functions are contained in a cluster, in a group of contiguous genomes, and these genes for what we need. So the interesting thing is that it was a genomic island that came from another organism, we don't know from where, but within this island we could find in light blue, you can see here, all of the genes associated to structural components of this secretion type 4 that had been associated previously in the transfer of genetic material that our organism in particular, we could determine that it was associated to the transfer of toxins from the bacteria that is producing it to the other bacteria. So a lot of analysis, molecular analysis to be able to determine this, but the consequences that this had, and the questions that we would ask is what are the ecological consequences of this? What happens in the bacterial community? So we carried out experiments where we could see that we had a biofilm of a bacterial that was sensitive to the system. So we started experiments in flow systems where we formed a biofilm in green, and then after 48 hours of formation, we added our competitor to the system. And what we can see over time in the top levels is that the blue starts increasing as the green becomes less, and that does not happen with the mutants that we place in the system. So, what is the ecological relevance? That nature, the sterile environments barely exist. So, if a bacteria comes to a place and at that place there are nutrients, there was a protection of secretions, etc., it's very probable that at that place there has been other bacteria before. Therefore, mechanisms that are successful in the colonization of different places have to use systems that will allow them to colonize spaces that have already been colonized by others. So we think that this system can contribute to that. And since this was a bacteria associated to plants, we carried out a series of experiments with different pathogens associated to plants to see the relevance to be able to publish where we wanted to publish. But the important thing about this, and this can be seen, this is a comment that came out in the when we published this article, especially the second point, that understanding the mechanisms of plant and bacteria interactions, not only specific terms, not only in bacterial communities associated to plants, but it can be associated to other kinds as well. It will allow us to understand the other principles that govern the successful microbiome by communities in other environments as well. So that was part of my doctorate work, and after that, when you are doing a little bit well, you want to go a little bit deeper, and want to increase complexity, and then you kind of, why did I do that? But anyway, we had a system where we could see how the microbes interacted amongst themselves. We could see it in the microscope, it was very pretty, etc. But one of the principles of the microbial ecology is that it affects what happens within the communities. I wasn't worried about the environment. I wanted to go more to the human part. So there the idea was, how can we use the same principles but now using the human body as an environment? In this search, we come to the MIT laboratory where the mucus was the subject of study. So literally, so the biogel that covers all of the humid parts of our body. So historically it has been described like this by different public functions. But I came here because at that time we were starting to realize from different sectors that the place where the microbiota lives in the different parts of our body is colonized by bacteria, except for the skin. It is a humid epithelium, and the bacteria live in this material that has different properties that we can discuss if necessary. So, what is important here? That the evidence suggests that there is a lot of diseases or illnesses that are characteristic because of the presence of bacterial infections, and in many cases, part of the microbiome that are capable of dominating the new community. And this disease happens where there is a deregulation in the production of mucus. A lot of mucus, not enough, anything, any alternate, any anything that alters the homeostasis. Therefore, the question that we asked was different to the question of biblical medical biology in general.
So how the organism is causing a disease. And what we ask ourselves is, why do we have these opportunistic pathogens where there is a specific population and they just get one person sick? You have virulence, toxins, etc. But what happens with the other people who don't get infected? Our general hypothesis was that a healthy mucus prevents an infection, keeps the microbial community in balance, and even when there's a presence of opportunistic pathogens, this won't cause a disease. Yes, that's an ambitious premise. I would work with engineers, but in order to do a systematic study with this, we needed to have an experimental approximation that allowed us to establish this hypothesis. And one of the simple ways of doing so, and the most significant one that they did, was isolating the responsible component of the viscoelastic properties of mucus. It's a glycoprotein called mucin, and you can isolate that, and then you can do a study where you modify the concentration, the pH, etc. We isolated that from an animal source, and with that native mucin or mucina, and I hope there's nobody has sigma here, but it's a lot different than that. But with those native mucina, we asked ourselves several questions. And the first question, based on my experience in microscopy, let's see how it lives in a gel of native mucins. Let's look at it in the microscope. We looked a simple medium, and we see that it grows, creating a biofilm, nothing in the beginning, and then it's a sediment. And then in the same medium where we added mucin, we see it grows consistently, and it grows as a cluster of added particles. And we cultured that with a window of time where we could differentiate those that we added. And the question we asked ourselves was, okay, this looks different. And what is happening with the pseudomonas when this is growing, the aggregated is growing? And what we did was analyzing the pseudomonas and opportunistic pathogens. And in many sites where you have different types of mucins, we chose two, MUC B, MUC 5B, MUC 5AC, and I'm not gonna get into detail with that, but what we did, and since we didn't know what was the effect of the pseudomonas, we asked a general question and we said, okay, let's do transcription studies, and we did so with two different mucins, and we saw how the expression pattern changed. In the graph over here, what you see here, each one is the expression of one gene in particular. And if you move to the left of zero, you can see that those genes, there's an expression of that negative, of the addition of mucins, and that diminishes the expression of those genes. And if we look at what happens specifically with the genes that diminish expression, and we group that in functional groups by using different analysis, we see that not all of them, but most of the genes that diminish their expressions are associated to different paths of virulence. This suggests then that what happens in the healthy epithelium is a domestication of these opportunistic pathogens. These are there, they grow in a 3D groupation, not associated to pathogenesis with this film. It doesn't grow differently, but it doesn't grow with the specific genes in order to trigger an infection. And this was something we proved in different levels. We used cellular models, animal models, infections. We identified that it wasn't the entire mucins, it was just a few sugars complex. Uh, and in general terms, what we described in the study, and we did so with other organisms as well, and this is that in a healthy epithelium, the presence of mucins contributes to maintain these opportunistic pathogens. We're not talking about bacteria that what they only do is cause infections. And we did with a non-virulent virulent mode. And whenever there's a change, such as stress, the physical properties of mucus changes, and that's the moment when an infection can occur. So what do we do with all of this? And since I arrived in Catholic University, the way in which I did all of this was that what we need to do is to continue studying this, but from another perspective. We need to try to understand what happens in the panel that we see to the right, where in conditions that despite having mucin and where there's a lot of mucus, there's a lot, there continues to be infection. And you may consider the results that I just presented in a situation where without mucin we have an issue, and with mucin uh, the problem is solved. But there are situations where there's too much mucus, and that's an issue as well. And what happens with that? And those are the challenges we need to address. And in order to use that, and how the components of the mucus in general, and how the components of mucus triggers an infectious process, what I propose is that we should study the group of diseases. Mucobstructive is a good model because in this case there's a lot of mucus. And this is a very interesting case because if you analyze the literature, and this has even been described as one of the models that has been best characterized an infection. I personally believe that this responds to the pathology and the physiopathology. We know what are the cellular components from the human point of view that triggers all of this, and this is a disbalance in the ion transportation, which causes a dehydration of the mucus that is in the lumen of the respiratory pathway and airway. And concentration increases, physical properties change, and the movement doesn't work, and there's an accumulation of material that's absolutely different that and that wouldn't be there in natural conditions. And then this situation in events, situation that where we don't understand how this is happening, we might have colonization. There will be microbes coming from aspiration as well. And what we know after 5 to 10 years is that a person is coming because then they have limited respiration, and that's because there is an infection that has been there for a long time, sometimes on a period of time that cannot be determined. And we don't know why this happened, and we know about it in general terms, and we don't know what's the initial event of the infection. And we can focus on the structural components of the mucus. A component that wasn't considered enough was the extracellular DNA. When I talk about this, I talk about DNA as construction material, which is abundant in that environment. And this is important because we know that in some samples of saliva, sputum, and there might be concentrations of 0.5 or 0.4 of volume, and in healthy conditions, it's very difficult to find evidence about this. Most of the evidence of healthy people is people who wants to do studies of microbiome, and they have the issue, and they need to get rid of the human DNA that's floating around. But they move in this range of concentration. And since I spent so many years working in a gel lab, what you can do is ask yourselves, what happens with the DNA in these conditions? And how we establish this plan, this this we're always working with that, and we want to work with concentrations that low enough so that these don't ruin our PCR, right? So the DNA in the range of those concentration is basically liquid. But if we increase the concentration and we go to the lower range of what characterizes disease condition, we see the DNA is something absolutely different. And if we have 0.5%, we know this is a different material. If you have 4%, and although it hurts us because it's expensive, the question here is the environment and the fact that it is absolutely different. And now think about this. Think about this. This bacteria that lives in this liquid, nothing changes. And when you are in in a situation where the geometric conditions are absolutely different, and what we propose is that this will cause changes in the behavior. And we were lucky enough that those arguments allowed the reviewers to be convinced, and hopefully ANID will release the funds to continue the work if that happens. But the idea is to start studying this to understand how it grows, specifically in this the Pseudomonas aeruginosa in this environment. What we want to do is understand a phenomenon that I believe is very interesting and significant, which is how the bacteria grows in conditions dominated by extracellular DNA and how these are tolerant to the antimicrobian. And I'm going to stop here for a second because the tolerance is a process defined by bacteria that should not be resistant. You have a minimum uh, concentration, the patient is sensitive. If you give too much antibiotics to a patient, 2, 3 days, they continue to have the bacteria. You isolate that, but that factor continues to happen. And when you don't know it, people talk about it, tolerance, resistance, etc. But what we know is that when we have bacteria that is not actively growing, and that's in a 3D aggregated um, and that can be resistant to the diffusion, and when it's forming biofilms, it ignores the antibiotic and it has the synthesis proteins and it's not it ignores it, and that's what we need to understand what happens there. We want to characterize the pattern of expression of the population of the tolerant population so that we can later develop alternatives and of things that we propose that we think that we can break the DNA wall, we can affect the structure of the biofilms, we can revitalize the bacteria that's laying there. And that's potentially something that we would like to develop. And in the last 20 seconds I have left, I think that one of the challenges that we have ahead in the future for all of those who are working in with bacteria is considering how the microbes that have this skill of creating this biofilm, whatever it is, in the environment, nature, or the hospital environment, how those microbes use that strategy, use that tool to cause infections in the patient. And the answer to those questions will not be by finding a microbe associated to plastic and asking if it has resistant to cephalosporin of fourth generation. What we need to ask those microbes is, hey, how did you got here? What have you eaten? What happened? How do you do when it's cold? And when we understand this, and when we understand that this is something that the microbiologists and microbians have learned, it's something they have asked their entire life, we can do it to these different individuals. We will be able to close that cycle, and we will try to avoid uh, in hospital infections, and that's something that happens for when we have bacteria that live in surfaces of the hospital, and if we are capable of eliminating that, we will be able to overcome this issue. And I feel really bad because I included a slide where I'm thanking everybody. I didn't include that, so thank you. Thank you, Gerardo, for the presentation. Do we have time for a question? Yes, we have time for one question. Thank you, Gerardo. Very interesting presentation. And I do have a question, and I'm not sure if the about the approach of the Pseudomonas. And because Cordelia was a Pseudomon, classified Pseudomon, it could have the same approach of the way we, or the way that we approach it in, in the hospital when it's already affecting the patients. When you mentioned the mucus, that's think that's something very interesting as a preventing option. So if you have any different approach, can you give us some more information on that? Thank you for the question. I think that I yes, I did work in a doctorate, and it was a strategic decision of my old boss. He wanted to make a difference from those who were working with the pheromone. with these pathogens and the pseudomonas. And my approximation is to first use the model that we know most about that has available tools, and there are plenty strategies of environmental pseudomonas that you can use. And then the natural transition for another negative basilicum, and from our point of view, allow us to do those similar experiments to answer those questions. But one of the advantages of working with the pseudomonas is that most of the tools, molecular tools that you use with them can be extended, almost all of them can be extended to other gram-negative elements, not necessarily in the intestines, but it can be used, yes. Any other question? Yes, we have another question. First, I'd like to congratulate you. It was very interactive, very interesting. The new information you provide with this work. And something that was very interesting to me is when you talked about cellular DNA. I'm not sure if there's any kind of biomarker that you have used to make sure that this is a human, because in those same organisms we can have we can find others that might be confusing. That's a really good question. And for a long time, we assumed that with if in the context with no infection, there was that cellular approximation. Yeah, because these are cellular networks that get to that area, project their DNA, and that's what's being accumulated. But with other studies done 5 years ago that identify the origin of this extracellular DNA that was found, and what they found was the highest contribution were eukaryote cells and not necessarily extended networks. And what they are proposing is that in this site, there are neutrophils trying to fight this, and this produce illicit, and this DNA is accumulated in time. And since the lumen is constant for a very long time, it can reach the concentrations that we found. There is bacterial DNA, but at this level of concentration is less abundant. All right, thank you, Gerardo, for the presentation. Please, Boris, head of the department, come forward. He will give you a gift from our institution for your participation. Thank you, Dr. Cárcamo. We are a little late, uh, and so the coffee will be at 11 a.m. and we will continue. They say the coffee is 2 for 1, so please stay and wait until 11 a.m. And we will continue with our next presentation called Analytic Chemistry as a tool to do census in our environment. And Dr. Fuentes from the University of Chile will be in charge of this. He's a biochemist of the Catholic University of Chile and science doctor specialized in chemistry from the same university. He is also a doctor in microbiology of the University of France, and I studied this last night. I practiced my French. Yeah, a little close to it. According to my translation, it's a doctor in microbiology of water and chemistry and mycology of the University of Po. The investigation line is called analytic chemistry associated to organic in compounds in the environment, and he studied the presence and behavior of these organic compounds in the environment and in foods. He considered the development of analytic methodology and anthropogenic in environmental matrix and diverse foods. He has been focused on the assessment of environmental markers and chemistry markers in agricultural products associated to immune analysis in antibacterial uh, products in bees. I give the floor now to Dr. Fuentes.
Thank you very much for the invitation to carry out the presentation here in this activity in the Scientific Conference of Public Health Institute. I am going to make a change regarding what you have heard about until now that has to do with microbiology. So now I'm going to go into the chemistry part, especially in analytics of chemistry. Why did I place this title? Because analytical chemistry allows us to do quantification of multiple substances in the environment, whether it be food or the environment. And it also works thinking a little bit about how we can deal with the environment or the in different senses. So we have tools to be able to survey our environment. And in this case, analytical chemistry is a means that allows us to do so in the food, in the environment, etc., in different disciplines. And that is why here we have analytical chemistry as a tool that allows us to interact and evaluate, for example, the climate impact. And this connects somehow having information to be able to study what climate change and health is at this point. And this is also what we're all talking about today here. So we already mentioned what we do in the laboratory. Our laboratory is the analytical chemistry, and we're from the University of Chile. I'm not going to go into a lot of detail about what we do there as a line of research. So going into the point, we have been developing different research in the laboratory to be able to develop this analytical tool to be able to have a quantification of different substances in the environment, starting, for example, with the determination of different pesticides is in the soils. Why? Because you know that due to the industrialization of agriculture, due to the demand of food, it has gone also in the increase of the use of pesticides to control the plagues that are affecting the different crops. And these pesticides affect the soil, and the soils are a reservoir of these components, and that is why it is interesting to be able to quantify them. So we have been developing a series of tools of analysis, meta-analysis, to be able to have a quantification. I'm going to name a few of them here, which have to do with the treatment of the sample to be able to separate the compound from there and then be able to quantify it. For example, we can say at some point that what you can see here, the possibility of applying microwave to be able to separate or extract the pesticide of interest in this time it was one from the floor, the soil, and to be able to clean it. Because when you take the pesticide, you also have other compounds that we are not interested in or that they bother in the quantification. So the idea was to be able to have an extraction and a simultaneous, and that is what we were able to accomplish in this drawing that you see here, where there is a microwave glass and where we can see first the extraction, and then we have a simultaneous partition to be able to separate and then to quantify it. And then that way we were able to have a certain amount of compounds, as you can see on the list, that are organ chloride compounds that are in a soil of different natures with material compounds. That is a factor that influences to be able to carry out a matrix that is pretty complex to treat. So then we also need to have a determination, and we change the structure, and there we have a different kind of interaction. So we have to change our strategy and change the extraction method and include, for example, a phosphate salt that you can see here that would allow us to extract that compound because there is a change in because of the interactions that are mostly on the surface and different absorptions, and that way we can improve the separation and to be able to extract the compound from the floor. That allowed us to quantify these compounds and be able to later study, and this gives us tools available so that somebody can use them and quantify the necessary quantification. So now let's go. We have been let's look at the organic contaminants in food. So we started, for example, studying the presence of pesticides in olive oil. Why? Because the olive is affected by the attack of a fly, which is the olive fly, and to control it, we have phosphorus pesticides. And what happens is that to be able to obtain a liter of oil, you have to treat or from 5 kilos of the paste that is made of the uh, olive. You made a paste of it first, and then they go through centrifuge, and then you have 1 liter of oil. So out of 5 kilos, you get liter of oil because you're concentrating the compounds there. So these compounds are liquid-soluble, most of them, therefore they can be found in the oil or they could be found in the oil. So what we did once again was to develop a method to be able to quantify these pesticides. We had the experience of having used the microwave, so we used that again to be able to assist in the extraction. And then once again, we did the partition liquid to liquid from a liquid sample, which is olive oil, to an extract through a solvent. Then we had cleaning that you can see here, which is an extraction process to be able to remove some interference, which was the oil that was co-extracted. Because with the compounds of interest, we could have something that we do not want to look at, and that could be an interference. So we had to eliminate, and that is how we eliminated, and we were able to have the we could do the whole cleaning procedure. So we saw that there was a limitation there that the glasses had to have specific glasses and they are expensive. And then we had during the process we had different complications. So we wanted to do this in an open system, not with a lid that was sealed with pressure, but we just used a different system in a laboratory that was much more simple. But somehow we had to avoid the solvent from being evaporated, because if you put a solvent that can be that can we had to avoid for it becoming vapor or condensing. So we generated this little refrigerant that goes over the glass so that we can avoid evaporation, and that way we can have the extraction during the process, which was pretty short, and generate that therefore the sample for analysis. This was optimized through a methodology called super response on the surface, and that way we could also create a method for the quantification. So once we had this method, we went to determine or analyze a series of samples of the oil that is created in different parts of the country. As you can see here, these are samples from different valleys where we have oil production, like Copiapó, Aconcagua, among others. And what we found the most and the highest frequency was of chlorpyrifos, and the second place was diazinon, and the low concentration levels. But at that time, there was something that the maximum limits allowed of olive, and but not in oil. Probably would be the maximum limit would have to be 5 times bigger due to the concentration that is produced. But what caught our attention was the presence of chlorpyrifos. Why? Well, not so much, but this is one of the pesticides that is the most sold, according to the registry of SAG, and used especially in the 6th region of O'Higgins and in the region of El Maule, because there's much more agriculture, therefore there is more use of these pesticides, and this can be reflected in the samples. So we can see that these patterns are repeated. Here you can see that once again you see the study of the different samples of different parts of the country. These are from the northern area where we have chlorpyrifos in concentration. And here you can see the samples in the region of O'Higgins that you can see more chlorpyrifos in higher concentrations than other cases. And here we have from the region of El Maule, where you can diversify the type of contaminant. Not only do we find chlorpyrifos, but we can also see presence of diazinon and other components. There you can highlight the presence of chlorpyrifos in the samples of the region of O'Higgins. So then we went to study another series of compounds that are denominated, they're called polyclinic aromatics, and they can be used, for example, or you can find them in food that is smoked or in the barbecues because you use the coal, and the coal can maybe not be burned completely, and it generates these compounds that is going to affect these meals. And also in oils. Why? Because there are some oils that are used not from the first press, but oils that are refined. So there you can also find these components. So what we did here, here once again, we're looking for it in olive oil. And what we did was see a different methodology, because what you do here is to try to innovate regarding the tools that exist, because there are tools. There are tools that are much more specific. For example, HVLC with a mass detector, but they are very expensive. If you know what I'm talking about, it's many hundreds of millions of pesos. So alternatives are always interesting, and in this case, the alternative was to design through fluorescence, a very sensitive fluorescence. So, due to the structure, as you can see here, due to the structure that these compounds have, they were fluorescent. So what we did was develop these methods based on fluorescence. And the first thing that we did, well, this was a part of the doctorate work of Francis, who is here with us from some time ago. And what we did was develop this method based on fluorescence. And the first thing that we had to extract the compound, but there were things that interfered with quantification. Sometimes you propose things, and on the way you realize that problems come up that have to be resolved. That's part of research. So here, for example, this that you can see here is a spectrum of uh, that has compounds, and as you add, you can see that there is no increase in concentration. You would think the intensity would increase if I add more, because there was an interference in the middle, which was the chlorophyll, that would not allow us to carry out the determination, because it would absorb the fluorescence that they emit. So there would be an internal filter, so to say. So we had to eliminate chlorophyll through a treatment of the samples. So once it was treated and that was eliminated, there we could see a proportional signal. So once we resolved that, we went to the quantitative part. In such a way, we were able to use these spectra to be able to carry out the quantification through what we know multivariable calibration that I won't go into detail, which is used for quantification. So with this quantification carried out, what we did afterwards was to also try to generate a method that was more sensitive, to be able to put it on a membrane, deposit the sample on a membrane, and then there they fluoresce, they become fluorescent. And you can see, for example, you have a sample of white that does not have the membrane by itself. Then we have the membrane with presence of the compounds that are on the surface, and you can see the fluorescence, you can see the color. And the other one is a sample of oil treated that does not have the compounds, and then in the end we have a sample of oil that does have the compounds. So we see the presence of fluorescence, and that is measured on an equipment, and it generates these that you see here. And this is translated in a matrix of emission that can then be used for the quantification. Instead of using the other method, you can use this one for this determination. And what we obtained was the presence of some in a very low concentration. We're talking about parts by billion, by kilo, of components. Like, for example, you can see here benzone, tan, chlorampheno. Some of them had dibenzone, anthraceno, etc. The levels of concentration were very, very low, but they were present. And we have to think that these are photographs, so to say, of the samples that we're taking in that opportunity. So, what we are proving is that these compounds can be found in this kind of food in very, very low concentrations. And now we're going to another kind of food. which is what we are doing more recently in the laboratory that has to do with the honey and bees. Bees can work as environmental sensors since they can move from where they have their beehive, forming their community around 1 kilometer if there is enough flora to be able to feed them. And that means around 300 hectares of land. But when there's not enough food, they can go up to 3 kilometers around the beehive, which increases to 2,800 hectares. Now, if it is much more serious, in critical conditions, they can go up to 12 kilometers around the beehive, which means a surface of 45,000 hectares. And what they do, the bees, is that they pick up the nectar visiting flowers, and those flowers can have some contaminants that then the bee assimilates, and then they end up being showing or uh, basically telling us what they have in the areas they visit. So we can see pesticides in the bees. And we can also see that we carried out some things in honey, understanding that it has certain properties, and it's always interesting to study. So one of the pesticides that we were looking for was imidacloprid, which in some countries of Europe it's prohibited because of its negative effect on the population of the insects that pollinate, especially the bee. So we did the fluorescence. We had learned how to do it with the pH. Therefore, we said let's try this with this compound that is not fluorescent per se, but if you break the molecule and change the structure of the molecule here, you can make it fluorescent and you can quantify it. So first we did a test in water to not have it so complex as honey, and it does work in water. So then we tried with different details that have to do with the analytical aspect, and then we went to the bees themselves. So what we did here with the bee was have a sample, a sample from the bees, and carry out like a paste in a phase that is C18. We had to extract the components and put it in this phase here in this column, and this is dispersion in solid state. So then you wash it with a solvent to be able to eliminate any fat or any other things, and then we dilute to be able to reach what we are interested in, which was the imidacloprid, and so we were able to see it with two different methods to be able to contrast and see if it was giving the results that we expected. Once we carried this out, we carried out the analysis of the samples of the bees that were collected in the sixth region. This is the places where we got them from: Rengo, Chimbarongo, Codewa. We did not find this pesticide because the concentrations are very low. Therefore, it also has a time of permanence, and then it cannot be found most likely. But we did carry out the exercise, and we did not find it. However, we went to find other pesticides that are used in more higher quantities. So we used the method that we had already tried. As you can see, we did the quantification once again, and there we looked for compounds that were organochloride and others that were organophosphorate. And so we did this with the different natures. Um, we did that with FPD, for example, and we got samples from the sixth region, and we added from the fifth region as well, in the region of Valparaiso, from the different areas that are in the red circles.
And what did we find? Chlorpyrifos, once again, we had found that in the soil, in the oil. Now we found it in the bees. This means this is a ubiquitous compound because it is used so much for plague control, as you know, and the sixth region, which is the area that we see it the most. So it's ubiquitous, and we can confirm it's present there. It can be found in different environmental compounds, and the pattern is repeater. That's a sample of the sixth region. When comparing what we find in other areas in the world, we indeed, this is the work that we're presenting, but there are some samples of other areas such as France, Egypt, and these are bees that went out to collect pollen, but have different kinds of component. These are forages, bees. In red, we include the bees that we found dead near the hive with high concentration, and you can compare those in red that have higher concentration than the other ones. So these are different things. What was the thing that we found the most? Chlorpyrifos is the one that we found the most. Second place, acrylonitrile, not something that we find as often, but you can find in some beehives. And because this is for the ácaros, it's very dangerous for bees. And when you compare this with the volume of sales of these type of plaguicides, the first ones are the one that you see the most in the fifth and sixth region, and then acrylonitrile not as much. Now, about honey. Honey is interesting specifically because of its characteristics. We know honey has been consumed for a very long time, and because of its beneficial aspects for health. And among the properties, we have its nutritive, it's nutritious, it's good for cancer. It has a lot of properties that make it special. Now, what's the secret of bees and why does it have these properties? And that's basically because of the chemical uh, composition it has. And one is vegetal component and another is vegetal animal component. First, we have the pollen and nectar, and it's that's what taken to the hive to make this the honey. Depends on the flora and the geographical area the bees visit. So what do we have? Carbohydrates, saccharose, um, starch that's break to create glucose. Uh, a third of the protein of the meal of the honey comes from the nectar phenolic compound that gives it antioxidant properties, some vitamins as well. Specifically, what honey has is sugar. About 80% of honey is sugar. The rest is water and smaller amounts of the other things that I just mentioned. So we would collect the samples of honey of different areas of the country directly from the producers or sometimes in fairs or like farmers market, not from the supermarkets, so that we could make sure that was pure honey. And phenolic compound, as you can see over there, it's a broad variety that you can find. This is a sample of the metropolitan region. Regarding the elements, you can find iron, which is always high concentration of copper. The cortex has much more concentration of iron than copper. But it's interesting to find copper because our honey has higher copper than the honeys of other countries, and that's natural because we are a country that produces copper, and of course our soil has much more copper. It's a logical sequence of why we have more copper in the honeys of the samples of honey that we have in our country than other countries. The other component is the animal component, and this depends on the genetics of the bees because that's what they contribute to the honey. And what these bees add to the honeys, it's proteins. Two-thirds of the protein that honey has comes from the bees. These proteins are enzymes, which is vertasa and defersin, and it works more with the gram negative, I think, if I'm not mistaken. And then glucose oxidase, which is an enzyme as well. And depends on the peptide, this is an antibacterial peptide of 51 amino acid residues. And the oxidase glucose that catalyzes the oxidation of the glucose in gluconic acid and oxygen peroxide. And this component generated in the milk is the one that, in the honey, I'm sorry, is the one that has the antibacterial properties, and that has a acid medium which gives us a pH of around 4 with high content of sugar with antimicrobial peptide, uh, causes this honey to be more difficult to have more bacteria, although it has a bacterial population that's quite interesting, but it's something from the honey. They're used to the honey, but that avoids other microorganisms to grow in it, which is why honey has an indefinite duration if it's stored in an area in an area where there isn't much moisture. It can last for years. The samples that we analyze with contents of proteins are within ranges that you can see in other places. But what is interesting is that the samples that are from more isolated areas, as you can see, a sample from Easter Island or a sample of Puerto Varas, and more to the south, have less protein content than if compared with the samples from the central area or the north. Regarding the concentration of oxygen peroxide, we see the results are very similar to what we find in other areas in the country, or the other countries in the world, compared with these areas in the country. It's very similar. And when talking about the generation of peroxide, we see that generation of this component is very similar if compared with the samples of the metropolitan region. There is a mechanism of the hydrogen peroxide that depends on the enzymes, and an aspect does not depend on the enzyme, and it depends on the flavonoids that are part of the phenolic compounds. So what we propose is a mechanism that includes the generation of it through the enzymes, and some has to do with inhibitions, fixations. There are several things that are part of the environment that can create this hydrogen peroxide. And lastly, the other interesting protein, which is defensin, and the first thing we wanted to do was extract it from the matrix and do with fluorescence. And why fluorescence? Because of the structure of the peptide. That in the extreme and the on the outer part C, terminal C, which is why we propose it to conclude this way. There are very few methods to determine this, and one of them is done through immunoassays with polyclonal antibodies. One alternative is an analytic model based on the models that we have here to do the quantification. With this method, we did an analysis of the samples in different areas of the country, and as you can see, there is a sample that from the Maule region and some areas in the north of the Elqui Valley. And the samples that have the most are the ones that are isolated, such as Sangaroa and Easter Island, because here we find bees that are associated to genetics and the resin that's created by the bees in their systems and products, in the immune system. And these immune systems are present, it's not adaptive. So this means these are populations of bees that have less defensin because they are less exposed to stressors, and that means that they don't need the same medium to control these stressors that might affect them. The one from Puerto Varas, we also find that it's very little as well. As a conclusion of everything I showed you here, we have the analysis chemistry, analytical chemistry that supports health regulations and environmental regulations. It supports public policies of food safety and sustainability, and allows to make decisions based on traceable and trustworthy data. And it also contributes to the development of clean technologies and safe food. And I have just a couple of seconds left, and I want to add that we just recently, in a work that we did in the context of a master's created by Marcia, We did a method to quantify the vitamin D in the uh, flour. And this is something that we did with Marcia, and she led it. I led it in the department at the university, and here it was led by Luis. And once again, thank you to the Institute for inviting me to this lecture. Thank you to Anid for the projects that provide us with funds to do these studies. And thank you all for the attention. You can ask the questions during the coffee break. All questions for the coffee break? Here, yes, we have a question. Can you hear me? Yes. A very interesting presentation. Thank you. I work in microbiology and analytical chemistry, and I have always asked myself how difficult it is to detect components, not just detecting them, but isolating them, in vitro or live. And in this case, do you have the experience of isolating copper from the honey? And do you have any possibility? Have you thought about isolating copper in that microbiome environment? This is related with the treatment of samples, and that means extracting what you're interested in, concentrate that, and then cleaning the sample and eliminating things that are not of your interest. And one of the areas that we work on very actively is analytical chemistry. And the data I showed of the quantification of copper, that's not so complex, because since you have to determine the entire copper, you digest or you use a sample, and uh, this is atomic absorption, depends on how you do it. But we have made more studies that I didn't include here, and on how it influences and how copper influences honey. Copper has a important role, because what it does, it's creates a complex with the flavonoids, and the complex that it creates gives a characteristic to the flavonoids that make it pro-oxidant, because flavonoid is antioxidant, but it can be pro-oxidant when there's copper. So copper kind of changes the way it acts, and it contributes to the generation of peroxide and to other properties as well. Copper is very interesting and very important in honey. Probably our honey, since it has more copper than honeys in other areas of the world, uh, I'd say it's special in this case. That's what we have seen. Thank you very much, Dr. Fuentes. Thank you, and we're going to ask the head of our department to give you a gift from our institution for your participation. Thank you very much. A round of applause for him. All right. We have a 30-minute break, coffee break. It's time. It's time for that, and it's time for the coffee break, and some time for you to check the posters. a calling for those who are in the coffee break. Let's continue with today's program. Today's conference is Climate and Health, and we are going to listen to the next conference titled Climate and Health Challenges for Chile, presented by Dr. Sandra Cortés from the Catholic University of Chile. Dr. Cortés is a veterinarian with a master's in biological science and environmental sciences and doctor in public health from the University of Chile. She is a teacher of postdoctorate degrees and a master's in public health in the School of Medicine and other faculties. She does her research in environmental technology and occupational technology, risk occupations affected by environmental changes and exposure to chemical contaminants, and studies of zoonotic diseases. She is the head of epidemiology of the Department of Public Health of the School of Medicine since 2012, and she's a member of the Commission of Masters in of Biology and Public Health. She is the part of the Association of Epidemiology and part of the International Society of Environmental Epidemiology. I leave the floor to Dr. Sandra Cortés. Thank you, Esteban. Thank you, everyone, for being here and listening our presentations from the Scientific Committee of Climate Change. I just want to correct something. I'm not the president of epidemiology, haven't been for a long time. That's a great relief because being part of the scientific committee would be impossible to do the two things. First, I'd like to say that in this presentation, I am representing the Committee of Climate Change, and I want to do a review of what we are doing in terms of climate action at this scientific committee, and I want to highlight some essential definitions and share with you some evidence and take a deep look into what happens with the climate and health and share with you what we are doing. I think this is very interesting because we are at a point where we are discussing a lot about the climate and we're putting the significance of the climate and health from the point of view of science. And to me, it's relevant that we all agree on the definitions, and some do not use the definition as we should. It's very likely that many of you looked at your phone and checked the weather for today. And we talk about the weather and the dynamics of the atmosphere in a specific time and place. And so what we did today was looking Ñuñoa, April 13th, and the information that we obtain is has a specific scale of time and place, specifically on the scale of days and hours. And the information it's just for 2 days. And that's right. But when we talk about the variabilities of the climate, we talk about how it changes and how the atmosphere changes at a space unit that's a little bit longer than just the 2 days. And it is important to consider this because that is related to the variability of nature itself and the variability that we are talking about today, a variability that we have caused as a species and that we have caused in the changes of the atmosphere. So this temporary window, it's from 3 days to 6 months, maximum a year. But when we are talking about the climate, we are talking about this predominant condition in the atmosphere in a time and specific place, but the window is at least 30 years. The 30-year window is what we call climate normality. That's what's normal about the climate of Chile, of Santiago, or for a specific commune. So when we say that we are studying, I am sorry because I can't really see the screen. Can I move this? When we talk about that we are studying climate and health, we need to be specific about the temporary window that we are working with. What's climate change? What we're doing is we're talking about this relationship of the change of the climate that's attributed directly or indirectly to human quality. And there is this natural variable that's uh, representative of what's happening all the time of the atmosphere, but it's associated to a period of time of at least 30 years. I'm going to move like this so that I can see. All right. It's important to consider that when we are talking about climate or climate change and health, we are talking by default about a 30-year period of time, which is what we want to assess. And this isn't the data that we always have. And I would like to highlight the definition of adaptation as the adjustment to the climate, to the real climate or projected climate. We work a lot with projected climate so that we can understand adaptation as all of the processes that lead us to understand and process these changes and also take advantage of beneficial instances. And this is important because we are always talking about the word adaptation, but we should consider it and having our perspective is that we are talking about how to moderate and take care of the damage and take advantage of what's important. And when we talk about climate risk, we talk about the model of risk associated to this climate risk. So this is related to what we are talking about when we talk about communities, a population, ecosystem resources, but all the time the resource is it's an integral uh, it's equation where we have the whole vulnerability, and if you pay attention to this interface that we have here as the dangers that we have due to climate change and the conditions in which this exposure occurs and the foundation of why it happens, it's modified by economic processes, the measurements we take to adapt or to mitigate, and it's affected by the governance as well, which is how do we organize as a community to face these risks, while assuming that the natural variability and the anthropic phenomenon will create emissions that will turn into the dangers that we are facing on our daily lives. And we are talking about mitigation when we talk about all of the interventions that we do to reduce emissions or to improve these options where we these things where we try to get grab that. And we're talking about the exposure of a community or a person, and when we talk about an exposure that is not just negative but might have some beneficial factors such as social economical conditions or the place where we live or the patterns of the activities that we do. And vulnerability is the degree in which people and system can overcome adverse effects of climate. We also talk about resilience. This is something that we discuss with our colleagues from other health institutes. And when we talk about resilience, we're talking about the capability, whatever those are, of the social systems, economic and environmental, and how do we face them? How do we organize ourselves so that we can maintain the main function? When we talk about the resiliency of the health system, We are asking what can we do for a health system to continue to work and doing what it has to do so that it maintains its identity, its structure, but also is capable of adapting, learning, and transforming. So, the concept of resilience is the most important concept that we should manage or use when talking about climate action because it tells operate, continue doing what you're doing, don't lose the identity, and prepare and get ready for the changes that are going on. With that in mind, I want to show you evidence, and this is something we prepare, and it might make you lose your hopes. So let's listen to this. Let's try to recover from this. And this is something that I really like because it's something we used when we did the first adaptation of climate change in the health sector in 2013, and we finally used it in 2016. We had two scenarios. The scenario on the left, which is the scenario where we used to do everything that we could to reduce CO2 emissions, and the scenario on the right is what happened if we did nothing. And the first figure shows the temperature projections, and the other pair, the rainfall. And let's look at what happened. We didn't do much. We didn't do much, right? And we have a specific area where there is an increase in the temperature in the highlands towards the mountains, and we have variation of temperatures in certain areas of Chile. And we have an increase of marginal temperature of zero point 0.5 in the area of Magallanes, projected from 2016 to exactly what's happening now. And in the figure of the projections of rainfall, if you see the area of central Chile, an area was going to have drought and an increase of rainfall in the coastline of the highlands and an increase of rainfall in the austral end of Chile, which is exactly what has happened. We also have a lot of information, and for those who like maps, look at the Copernicus map because Copernicus has several images that are really valuable and are they're in real time almost for several things. They have greenhouse gases, fires that we discussed yesterday. This is a figure of November 2024 where we had a concentration of fires and the radiation power of fires in the area of Africa, the Amazonas, and the Central American area, and Central America, Mexico, also Chile that has a lot of fire activity. And this is a slide, I mean, this is an image of when I just started setting up the presentation on May 6th. Chile, and despite the fact that we are in fall, continues to have some fire spots, and it changes. We know this is dynamic. It's very interesting that the evidence in Chile, and we have this publication of 2008, we have Professor Luis Fuentes from the Catholic University who analyzed the characteristics of mortality associated to the climate. And in 2008, that is 15 years ago, we already knew that there was a high risk of dying on hot days, specifically for the elderly, for the people who had a lower educational background, and it depended on the cities as well. This was based in Brazil, Sao Paulo, but and this is something that we already knew about mortality.
So, in this study that we carried out some time ago with a student that was in exchange, Rebecca, and here we analyzed fires in the central part of Chile and the impact in the children, and we could see that they had more bronchitis or respiratory issues in children, especially in Santiago, due to the bad ventilation situation. But in summer, when the quality is not working so well, when we do not have tools to handle that, and when the system was relaxed because the respiratory problems, we understand that it only happens during the winter, but we did find an increase of the risk of 1% multiplied by the 2 million children or 3 million children in the central area of Chile, which is a very important load in health. This other article, I like it very much. And here you can also see a collaboration of Chile with other countries because you can see the increment of the risk by arbovirus. And 2 years ago, it was said as an emergency in public health, which still exists. We know that there are people here present regarding the presence of the mosquito Aedes aegypti, and the probability of having the mobility of dengue, especially in the central part of Chile, knowing that we have other areas that are warm, as could be in Arica. Another article that we carried out with another student regarding the carbon footprint of Chilean diet. We have the consumption of food, and we were able to establish the footprint of the food cycle of the different food, and we were able to establish that the consumption of meat and milk contributes to the greenhouse gas emissions, especially in the south of Chile, considering the cycle of production until we end with the ingestion. And we saw the highest emissions of the GEI associated to diet come from those who belong to high social economic income, especially in the urban and usually in the center northern part of the country, and they have more access to meat, therefore they have more emission having to do with the diet. They're highlighting that not only is there an importance of the diet based on plants regarding the diet based on meat products, but at the same time that it can be sustainable, and that is a challenge that we are not talking about so much. This other report, this other publication, I find that is very important regarding the reduction of the coverage of snow in the Andes in the center part of the country. We're talking about a reduction of 20% of ice, snow in the center area, and we do know that this is going to have important implications in the water production, not only for human use, but also for agriculture, energy, hydroelectricity. There are many factors that are associated to this, but we're talking about an important decrease of the sources of water in the central part of Chile. We can also see the increment on the dry days in the center of Chile as we had forecasted maybe around 10 years ago. It is clearly documented with the increase of dry days in the central part of Chile, which impacts it is you can see the impact of the mega drought between 2010 2019. So in this scenario, to zoom in a little bit on the health, I would like to show you a few things that are very basic, especially from the WHO, who has brought up this model to be able to understand how the health phenomena, especially having to do with the risk for health of these diseases that are sensitive to climate, can be related to these factors of exposition and vulnerability. Within what the WHO and its model for the world in general, so that the countries can take into consideration, it talks about the dangers in health associated mainly to climate, and we are talking about the extreme temperature events, those who are very, very high or very, very low temperatures, heat, the increment of the level of the ocean. Was anybody on the beach this summer? Did you see how there are areas where the coast is or the beach has been reduced with its normal fluctuations, but it is a phenomena that is happening. The air pollution, a phenomena that is hyper known for Chile up till yesterday. I don't even know if you saw that, but we had very high pollution levels for the health of particulate material up to 5 throughout most of Chile. Another important danger is the distribution of vectors and its ecology, the lack of water, and the reduction in the production of food. With the base conditions that the countries have in our communities having to do with demographics, we already talked about the structure of demographics with more elderly, as we have in Chile, the distribution of geographical issues of vulnerability, and we also have the biological factors and the health conditions. Just remember that we have a country that has an prevalence or an increase of many chronic diseases, bad social economic conditions, and the capability of the health system, as well as with gender and equity. And that is part of the list of indicators of vulnerability that the WHO that we should um, evaluate, plus other conditions having to do with the exposition. So how are the cities built? How is the health structured? What is the energy structure, the water provision infrastructure, the production of food, and the health system itself? So what are the diseases associated to climate? or the outcomes that are important associated to climate. We know that we have injuries, we have mortality due to extreme heat, we have respiratory, cardiovascular, zoonoses, and other diseases that are transmitted by water or through foods. We have vectors, malnutrition, chronic diseases, and the mental health and psychosocial issues. So the scenario is complex. This is a fantastic model for those of us who like complex models because this is the reality that we have to deal with, having to do with climate change and health. Numbers from the WHO, obviously the climate change influences in the social determinants and as health, environmental issues due to basic things like, for example, air, water, food, and where we live. We have 250,000 deaths having to do with malnutrition, diseases like palliatism, diarrhea, or caloric stress, and other that there are an additional 250,000 deaths with costs that go from 2,000 to 400 billion dollars. So when we have our objectives for sustainable development in Chile, this is something that many forget, but Chile has seven of the nine criteria of vulnerability associated to climate change: low coast areas, arid and semi arid areas. Then we have forest exposed to forest deterioration. We have the native production as well as the forest. We also have the territory that is susceptible to natural disaster. We're all experts in natural disaster in this country, at least those of us who have to do with environmental issues. We have seen many of them. We have urban areas with pollution, atmospheric pollution issues. The central region, metropolitan region, fifth region has 8 million people, 9 million people concentrated in the areas with many dangers having to do with the environment and air pollution, mountainous ecosystem, and areas that are easily affected by drought and desertification. Due to the climate in Chile, here you can see in this figure on the side, basically the cuts, the transsexual cuts. You can see the south green. It has a characteristic of geography and bioclimate that is completely different from the north or the center south of Chile. And that is great wealth of this country. However, it also has a very specific profile for each place. And this is what we see, right? This is a picture of the Maule 2 years ago. The fires, the industrial parks at the on the coast. We have problems with the coast and air pollution. So there is an important complexity in what we define in the scenarios of exposition that forces us to have a response where maybe the institutionality does not work because we have an institutionality that is very vertical, right? So what the norm says is for all of Chile. So this is some of the data from from the Lancet and the little bit of evidence that we have in Chile with data that tends to be secondary data increment of the hot days that children experiment under 1 year of age or 65 years and older, the increase of the loss of hours working. You have the surface that due to drought. You have death due to particulate matter having to do with the burning of fossil fuels and biomass. So, this is one of the main things. We cannot continue forgetting to talk about this. We need to find solutions for climate and for the people who are being affected by this. And also, according to Lancet, more countdown and the different reports are generated in this part of the country. So we have a lot to do still. So starting in Chile, Chile is one of the few countries who has a law having to do with climate change. In this framework law, they establish responsibilities in a national, um, regional, and local level with different actors who are with the Ministry of Environment. You have the different strategies having to do with climate in long term. You have how much Chile is proposing as goals with different milestones to be able to have climate action oriented to the drop of emissions in particular throughout all the paths that we can, plus all of the sectorial plans of mitigation and adaptation that all ministries in Chile have to connect to, especially the Ministry of Health, with a drop in the plans to the regions, and each region has to have a regional plan of climate change, and all of the different communes also have to have their own plan for climate action. Now that elections are coming up, this is a parenthesis. You can raise your hand and ask your candidate, what is your plan? What is your regional or your commune or your national plan, to see what they can say. And additionally, we have strategical plans of water use, recognizing the importance that water has for all the different rights that we have. And we also have the committee having to do with climate change, which is an advisory committee that is here by law. We had a previous committee that was due to the COP25, but we did not have a law yet. And one of the products of that scientific committee was to be able to reach out to be able to have this framework law. So we thank them for that, and that has been incorporated into the law. And many of the members of this first scientific committee today are main players. For example, the minister. We are the first scientific committee created in this mandate. We have 5 countries in Latin America with this one: Costa Rica, Mexico, Guatemala, Chile, and one more. I believe it's Ecuador. Brazil wants to have their own committee as well. This was open tender. We called, we argumented, they chose us, and among us, we chose the president. And I said I wanted to be president because I believe that from the scientific committee, because the evidence and the action having to do with health have to be priority. And if I was not there, I was afraid it would not be that way. So we have gender parity, we also have territorial parity, we have different disciplines, and there are quite a few people, and our only task is to ensure that all of the instruments be based on the best evidence, scientific evidence available. Chile has to, every 5 years, they have to have this contribution, which is its commitments expressed with goals, whether it be in the sector for each ministry. And here there is something that is very important that makes us different from other contributions from other countries in Latin America, because we define as a country in 2020 that there had to be a transversal pillar of social ecological transition that is fair, so that we can see the problems and the social inequity that there is in Chile. And that is why there is a series of instruments of implementation. And here you can see the commitments with the different greenhouse gas effects, and you have the reduction of methane and carbon reduction, and on different sectors, the plans. This is being updated. Some of you participated in these scientific dialogues for uh, climate action, and we are in this process now. We are waiting for the Ministry of the Environment can publish their last final version of the NDC, but to be able to give the scientific evidence that is available, we set up, this was an experiment that came out well. We had scientific dialogue for climate action to propose indicator, because that is the problem. We need to have indicators that are very specific and criteria for follow-up to be able to know what is happening. But in this complex model, we need to know how interconnections happen and how the plans and all the different actions work together towards one good, which is to reduce emissions and prepare ourselves better. This was very interesting. We had more than 240 people who participated. We carried out the work as well as possible. And in the health area, and the due to initiative, personal initiative, we had these previous meetings with the group that we called the Work Group in Health and Climate, where we have researchers, academics, the ISP, the school association or school medical association, to be able to capture other points of view. To the This is what we told the minister for the pre-project, and we wanted all of everybody to support us, so we needed indicators, clear indicator, robust indicators that will allow us to have early warning, that would allow us to monitor, who will allow us to modify and measure to modify the reality we are living. We said we had to have an ecosystemic focus that could be broad and intersectorial because we have to work in the different sectors because we need to have synergy because we need to work in the co-benefits of climate action. And we also requested to strengthen the territorial focus, climate justice, a human rights perspective with a view of gender equity, gender, and the vulnerable groups as well, that we already know what they are. In all of the policies of mitigation and adaptation that each of the sectors of the state and of the communities have to work on. I would like to close with this proposal from the WHO. The WHO says that this is a great moment for public health, for environmental health, and for occupational health because we have three axes. One of them is to have to face the impacts to see how much does it cost in deaths, how much loss in hours, how many people that are sick, how many cases of diarrhea we need to quantify associated to the climate variabilities in general. We need to promote all of the actions of other sectors who produce co-benefits in health. In other words, when we remove carbon as main element in the energy matrix and we improve in electromobility or we improve in the heating systems or we improve towards diets that are based on planets, then there will be co-benefits in health that we know that are going to produce positive impacts. And it is key to be able to work strongly with the resilience and decarbonization of health systems. We know, and you already know very well, or you should know, or if not ask, how safe is your hospital? What is the line, the flood line? Small things, but work for that in that direction. The next steps are, well, this is really not next steps, it's actually now. Today, climate change is making pressure on health, but we have not been able to make this health the truth in all policies, and now we have an opportunity to do so. We not only have to generate better and more research, but we also need to create dialogue. And here is a call from the WHO so that the health sector can become more of a public voice because when the public sector has public voice with evidence in its hands, locally, regionally, or nationally, internationally, we are contributing so that the different sectors can be involved. And they can be involved in a moment where we are promoting to not know evidence, scientific evidence. So the WHO is asking us to use this space because if we do not do so, then other voices will be heard. So we have to form the health teams. And I say in a humble way that I really believe that we are not experts in climate and health. We are experts in air quality, water quality, food quality, zoonosis. But to be expert in this means that we start forming people now, not for later. And of course, to increase the climate ambition, integrating the health pillar in all of the climate actions that can be carried out, whether it be nationally, regionally, and local, within the regions, within the communities, and in all of the different areas where it's possible, because that is what this time is demanding from us. So I would like to leave here the email of the scientific committee because we are open to all the concerns, questions, and we are open to be invited because I forgot to say that we are a group that works ad honorem. We are not in a budget, and therefore we are welcome to for all who want to collaborate ad honorem, obviously, as well, that can communicate and can be a part of this. And here is my email because in the end, the person who is seeing the specific focus in health is me. Thank you very much for to all of you for being here.
Thank you, Sandra. We have time for a few questions from the audience. No questions? Sandra, I would like to ask you a question. You mentioned indicators to assess climate change. And I think that's one of the most difficult things we can determine. And on one hand, I maybe not know other variables, but from the microbiology perspective, have you determined any microbiological indicator of the climate change? Indicators is a big topic, and there is a list of indicators proposed by WHO, and there's another list of indicators for last calm down, and I think we should have our own indicators. Of course, we need microbiological indicators because we need to check the quality of water, of food. We need to check changes associated to different pathogens, especially those that have microbial resistance, but not only to bacteria, because there are resistance sometimes to other products that are therapeutical. So I think yes, but we need to advance on making our own list. Of course, at the cut down link, it's very helpful because they have they organize it in numbers with three, four identificators, and I think they don't have microbiological indicators there. So maybe we can contribute to that. Thank you, Sandra. I'd like to ask a very a small question. Hi, Sandra. Thank you for your presentation. I am clim working climate, and I am very happy to work on this area. I am a meteorologist, so I want to ask you regarding the financing. Institutes such as me, do they have an agenda for applying to projects that are of the area of climate and health specifically, or nobody has talked about that yet? Thank you. Yes, the funding or financing for any public health project is key, but what I know is that we don't have any financing lines for climate and health. And actually, we have to prepare a report of the scientific evidence, and we are going to include a chapter of gaps of the investigation of the research. And of course, we want to ask for specific funding lines for open uh, contests to apply, but today, as of today, that does not exist. Thank you, Sandra. Please don't leave, Sandra, because we want to give you a gift from our institution. Thank you, Sandra. Let's give her a round of applause for her presentation. Now it's time for the next lecture presented by Mr. Diego Varela, and it's from Achipia. He's a veterinarian doctor from Universidad Mayor, master's in business, business administration and business in technological management with a focus on biotechnology from the University of Talca. From 2010-2013, he was an advisor of food safety, fishing, and agriculture in food health at FAO, and for the Caribbean as well, and AGECID, which is a Chilean Agency of Cooperation for Development. Between 2018 and 2020, he was the president of Codex for America and the Caribbean, and in 2022, he's the executive director of the Chilean Agency for Food Safety and Food Quality, and he has been part of the presidency since 2023-24. In his professional career, he has participated in several projects of international collaboration. I give the floor now to Mr. Diego Varela. Good afternoon. Thank you. And congratulations to the Public Health Institute for organizing the 17th conference. Can I move this? There it is, the scientific conference. So thank you. Greetings to the director and to the workers who are part of this wonderful institution that has had great collaborations with Achipia. the Chilean Agency for Food Safety and Quality. And the title is different of what I announced because I changed it several times. So it's called Science at the Service of Food Safety Opportunities and Challenges for Chile. Let's begin then by providing uh, a brief reminder of what science is. So the goal is to build the knowledge and understanding of the world around us by providing the foundation to face complex challenges. And the area that I am addressing is food safety, which means producing food in volumes and taking those foods everywhere so that they are available for people and they are safe. To have safe food, it's something that's quite complex and it's getting complicated with time. The science of science lies on following evidence, and evidence being the foundation of our conclusions. And that helps us understand certain ideas that we really like and care for, and that's what it does, science and decision making. Food safety as a scientific discipline. This comes from uh, the beginning of age, since humans are humans. We want to avoid food getting sick. And a long time ago, the science, this applied science, wasn't really effective, and the expectancy was around 25 years of age. And if people didn't die due to an attack of an animal or due to war injuries, would end up dying due to food intoxication. And what they would do is changing certain flavors for the stability of this food. And this science has been key for the development of humanity. In the 19th century, there were progress in microbiology. This was led by uh, people such as Louis Pasteur, who helped us understand milk and the understanding of the microbiological dangers. And that was a milestone in terms of understanding the risk behind intoxications derived from food. And technologies related to this thermometer. I will share with you some elements that have caused a revolution on this science. And in the 20th century, the discipline of food safety is something that is interdisciplinary because it includes chemistry, microbiology, engineering, statistics, epidemiology, and it is basically a science that gathers several disciplines, and we'll see it's something that um, continues to need this collaboration. In this global context where economy grows and the demand grows as well, science and the development of technologies and methodologies that protect food from physical contaminants, biological contaminants as well, it's the most important thing. And with no questions, we know we will need more science, not less, more science to produce more food that is safe. And in order to do so, we need to have agreements and to make these agreements as a society because we need to be capable of providing food safety. This is very important as well for social stability, political and economical stability of this country and the world. And some time ago, and until 2014, we were on a good pathway because we were reducing the amount of people that were affected by food safety. Was measured by the World Health Organization with specific indicators. But in 2014, there was breakage. The number that was going down started to go up. So thinking that food is something that we can guarantee, it's something that we should rethink. Of course, population has grown and we need to produce more food than we did before. It's estimated that double by 2050, 2100, with less resource, with less impact, and with less water. In a scenario in which climate change changes the triad of epidemiological triad and population growth, along with other demands that the population is having related to the foods that are very diverse, uh, if you were to organize a wedding, you would say, okay, you have normal food and then vegetarians because you would have a couple of them. Now you need to have a gluten-free, vegetarian, vegans, keto diet. It's very complex, and it's okay that it is like that. The growth of the population, along with aging and the people who are elderly, they are more prone to be affected by this, and those that are immunodepressed as well. In the past, they would have died. Today, they can have medication, but they are still very sensitive to the food that's transmitted by the diseases transmitted by food. This is also related to malnutrition. That's also pressure on us on having food that's safe for consumption, the growing urbanization and the changes of food feeding patterns, and the increase of technology in productive processes to produce food and what we call today as new food, such as those called plant-based algae, microalgae, which were things that we weren't used to consuming, food that come from fermentation and precision fermentation and cellular food. And we need to make sure that these foods are safe for consumption. And what's the most difficult thing to do, which is the communication aspect of it, to share these with the population. The waste of food, so in that trend, we start consuming food that we used to discard in the past, and we don't know how that's going to produce any impact in the long term. So if we start consuming parts of vegetables that we didn't consume in the past, what's the level of pesticides that these parts of the vegetables have? Same things for animals, and same happens with recycled food. So, as I was saying, some scientific innovations that have supported food safety based on science and scientific development, and behind this scientific thinking, where you can acknowledge what you did wrong, and not to hold on to these ideas. And it's very basic: refrigeration and freezing. And by the 20s, the capacity of refrigerating and freezing food caused an incredible change for the transportation, for the storage in big quantities, and also for the storage of this food in our homes. It diminishes the deterioration of the food, and it eliminates bacterial growth, eliminates parasites, increases enzymes. There are a lot of fresh foods that we could not access if it weren't for this technology. And then we have radiation of food. This is something unknown that might be controversial for some, but this is a process that consists on exposing food to ionizing radiations to eliminate insect bacteria and microorganisms. It is used with fruits, um, and leaves, and this is something absolutely sure, certain and safe. It is based on evidence, and this is a process that is safe based on evidence. Packaging in modified atmosphere, uh, vacuumed packaging is something that has helped us contribute food to last longer in better conditions. And the analytical techniques of high precision, such as spectrometry of masses, PCR, chromatography of gases. This allows us to detect contaminants and detect microorganisms, of tracking outbreaks and getting to understand what's happening. And that has helped us with this huge progress we've made. But there's a paradox. In the history of humanity, food has never been safer than it is today. And sometimes I hear, I hear some romantic uh, perspectives that food was more natural or people wouldn't get as sick in the past with the food. And this is a myth. Foods that are transmitted through, uh, disease is transmitted through food. And due to science, this has been possible, and that is because as a society we have agreed on all of this, and we have established, okay, science will be our base of decision making in terms of policies. And for all of us who are part of society and live in a city, we need to agree with different life projects, but we have these basic agreements, and among these basic agreements, science is our pillar to make those decisions. And I have to mention the food codex, and this isn't a technological advancement, but it's an agreement, multilateral agreement that used science as a norm, and almost 75% of countries in the world agree on that. And the Public Health Institute also participates actively in this codex, food codex, Codex Alimentarius of the Food and Agriculture Organization. However, we are at a different situation now where all of this progress is being pressured by a dynamic that we never thought we were going to have in the 90s or beginning of the 2000s, when it seemed like the world was rocketing and food safety was going down. And that is because we have a technological acceleration that we are not being capable of understanding, and as a citizens and legislators, even companies promoting it, are capable of understanding it. And these challenges require careful management and reflection on this, that we would want to do it more calmly, but we are not capable of doing so because these technologies that are advancing so rapidly are impossible to stop, such as AI. Although we had laws to stop AI, that's something that's not going to happen. I read a title the other day that about 80% of students of the first year of college of university are using AI, and AI started or had the boom of use in the past 2 3 years. So AI is being used, and also biotechnology. Both are making a huge progress, exponential progress that we never saw coming. And different than other technologies, such as the invention of the thermometer and refrigeration or freezing of foods, these technologies have some affectations that are ethical and practical. We need to think about these technologies, hopefully, calmly, which is impossible, but we need to adapt. This excessive dependence that we are seeing, that we're having, and it's more from our old school, uh, because we were raised like that. We used to look at books, encyclopedias, libraries, and now we that were raised like this are concerned about these new generations that are formed by AI, and which can affect critical thinking. When you're an expert in your topic, and if I ask the AI on food safety, I will immediately see the mistakes because I have a previous training. But what happens if I grew up thinking that AI, I mean, not looking at the errors provided by AI, I won't see them. And what happens then if we use AI on this topic, and it basically creates a black box no one would understand. Imagine having an error in an algorithm that would put in danger the entire food safety of the country. It could be a catastrophe. The Codex Alimentarius took decades to exist. AI, in a couple of ages, it's being used and starting to make decisions very rapidly. And sometimes we don't even notice. To get here, I took a bus. I didn't know which one to take. I used Google Maps, and it gave me the best option to get here. So a decision that I made on a daily my daily life was provided by Google.
The same in the social system that decides what to show us, the algorithm of LinkedIn that shows us what is the ideal job for us, etc. So we are going to need more and better technology to ensure the production of food, but we also need to continue with the dialogue as a society so that we can know and understand how we are going to introduce this technology. So science does not tell us what to think, they give us answers, but we have to be capable enough to know what answers we want to give. In this scenario, then, to be able to understand this scenario in a food safety, is that in Chile we have a hypothesis. We say that the national system of food safety and quality of the food is a social system. And in this social system, interaction between the members and competent authorities and producers and the chain and consumers has to be based on communication that makes sense. How can we make sense with these communication? This is something that is very complex, and you think about a food safety agency and you get into a more sociological theory. It is complex, but we have seen that it does happen. So how can we narrate, or how can we come to an agreement in what makes sense to Chipia, to the Ministry of Health, ISP, CERNAC, SAG, who have different roles, CERNAPESCA? What makes sense with the construction of a of a roadmap of a food safety that can be guided by a diagnosis, principles, by a vision and a purpose and objectives and strategic alignments. This policy is now under consultation. You can scan that QR code or get into the Achipia page, www.achipia.cl, and you will be able to click on that. Do not put a chipa. It is a chipilla because the other one is only potatoes. But this one includes much more than just potatoes. So we invite you to participate in the construction of this proposal that we are bringing up for the food safety for 2025 to 2035, where we are putting that together. We are coming in agreement to see what makes sense for the different players that are a part of this national system of food safety and food quality. So, any suggestion, or most of the suggestions that I'm going to make now, to strengthen the discipline of food safety and quality through science come from the dialogue that we have had here in the construction of this policy, in which, of course, ISP has participated. The first suggestion that we are always proposing from Achipia is the creation of a national system of food safety and quality to strengthen the capabilities of the institutions so that they can work in in synergy together and in a collaborative way to be able to go from working in silos to working as a network. The second, institutionalize the decision making based on the science. We have this declared as a principle, one of the uh, 10 principles affirming that the decisions that we make for everything having to do with food safety, which has to do with administration or management, regulatory, inspection, training, a laboratory of epidemiology, not have that's based on preconceived ideas, but to have it based on evidence, wisdom that comes after applying the scientific process. To implement in Chile the focus of risk analysis with its three levels. Um, this is a focus that is not very well understood yet, that still has a lot of confusion what risk management is, or risk assessment is. We still need to first make it work institutionally and then for it to make sense to the population. This focus is very well developed. For example, in Europe, Europe has EFSA, a European Food Safety Authority, that carries out scientific evaluations, only scientific, where they do not consider any other aspect, social, cultural, or political areas. It only does identification and characteristic of a danger, and then the characterization of the risk for consumption. That is the easy part because science many times is easier than interaction between the members and to try to have us all get along. Then we have risk management, and that is where we need to make the difficult decisions, where we have the recommendation of the evaluation, but we have to see if we have the resources. We have to see if we have the geographical possibility of implementing what is being said, if we have the political environment, etc. And communication, I would also say that is part of the most complex part that we have had to do. at least as executive secretary, to be able to communicate to people regarding food safety. I participated in an event where we were talking about risks that exist of diseases transmitted through food last year because it increases in you have more in Easter. We have more higher consumption of seafood and fish. We had all of these things that we recommended, and then what happened? The journalist said, "Why is the chocolate so expensive?" So everything is imported, and all the news, all they talked about had to do with the chocolate until, um, and the focus that we had on food safety. But what we do is more react until something happens. So our job is to prevent this from happening, and that is what they don't pay attention to. But in Chile, we need to place resources. We need to place them in the public opinion to have the resources that ensure that any person here in Chile or around the world, when they purchase a food in an established place, and there we can see with concern the relaxation that exists from the citizens to be able to consume food in non authorized locations, because when you have an established place, you can be sure that that food is not going to make you sick or, in the worst case scenario, kill you. We also need to know and understand and manage aspects related to biotechnology and artificial intelligence. This is the discipline. This, in other words, means to broaden the horizon from what we are used to seeing, which is epidemiology, chemistry, microbiology, etc., and to start incorporating these new tools. And that's why we need to attract talent related to artificial intelligence, with handling data. And I have seen that many agencies in other countries are starting to do so because they're automating their procedures. To strengthen this, of course, this comes from policy, strengthen formation and development of capabilities of human resources in the food safety and quality areas, in public services, in education, in the basic in schools, and to promote research that is applied, and also basic research. And as I was saying at the beginning, we need to continue thinking in a collective and critical way to why do we want science? Because if science goes over us and we start being a technocratic society, then we are not going to find a solution there either. We need to reflect about what kind of society we want to build and understand that science is a tool, that it's not the end, and that the dialogue that is informed, inclusive, and honest is always going to be necessary to be able to ensure that the food that we are consuming is healthy. Thank you. I don't know if there's any question for Diego. Hello, Diego. Thank you very much for your presentation. I really enjoyed it. I work with food, therefore, for me, it is very important to see what you showed. I would like to ask you regarding what you mentioned having to do with the use of artificial intelligence in the production of food. I don't know if the food codex somehow is in any of the if you committees, if you are thinking about how to do so. Nothing of that yet. No. The food codex is what the countries say it is. Up till now, no country has brought a proposal, and generally, it starts with a document that starts to create conversations regarding this, and then the FAO is going to bring one up very soon, and it looks very interesting. I've seen it. And in the codex, this generates controversy because the codex has 189 member countries. And when you bring up a subject like this one, the less developed countries or that have less income, they feel left out. and that is why you create certain tension. But it's a reality that has come to stay. During the pandemic, the Codex did pull up a norm or a guide for remote inspections, for example. So I wouldn't be surprised if it can quickly start reflecting regarding this. And they are also starting to see the follow up or the tracing. Chile could also bring it up. Thank you very much. Yes, having to do with that. I was asking that because Chile, in that sense, has been pioneer in the use of artificial intelligence with the experience of NACO that they use an algorithm to produce their food. So maybe that would be a good option that it could be that we could be involved in. Thank you. It would be very. This talk is very interesting. Another question. Hello, thank you, Diego, for your presentation. And I wanted to ask how to. We were also talking about climate change that we saw in the previous presentation. And from the agency, how are you looking at climate change and the quality of food? And how are you forecasting, thinking, for example, in the previous presentation, having to do with the temperature and the vectors, and how that also causes change in the dynamic of production, for example, in the agriculture in the country. So, what are you seeing? What are you projecting? How are you articulating? And what suggestions do you give regarding that subject? Climate change, when you talk about food safety, when there is a change in the epidemiology, everything changes. We start finding dangers in matrix where there was none before. Salmonella in food that before did not previously did not have that. Mycotoxins in geographical areas that never had that before, and those mycotoxins are very harmful in the point of view of those that are chronic. Something else that happens also with this change is that we start seeing different in how the crops are affected by certain plagues. So what happens there? What happens on the field? There's more pesticides. So it does have an incidence in maximum limits of residues. The same in the animal production. The use of veterinarian products also changes the dynamic, so we have to be taking a look at the maximum use of veterinarian medications. So we have a report that we put out last year that's where we can see a little bit the changes, and we use a lot of prospect analysis because to see it short term is not easy. So I think that we need to continue monitoring, and therefore it is very and important to be able to share data that the universities generate, that they have to generate for their publications, the IESP, the Ministry of Health, CERNAC, CERNAPERSCASRAG, to have all of that available in a database. That would definitely allow us. You always need more data. To the conclusion at the end of any study is we need more data. If we had access to that data, if we could be in agreement, I think we would have a lot more information. We would have a much better scenario, but that is a long conversation. And that articulation of the presentation that was presented in the policy of the intersectoriality, the databases are there. Therefore, to get an agreement in the different areas, and Achipia has a key role regarding food safety. So maybe we can start with that. That could maybe generate certain recommendations or previous actions, because what is coming is very complex, thinking especially in the access of quality. Most likely, there is going to be food available, but the quality of the food based on the new technologies and what is emerging is might be something that is complex. And we could already start acting as long as that we have this connection between the different units. The institutional logic adapt to all of this because the Ministry of Health is more decentralized, with the SEREMISAG has a centralized system as well as Cernapesca. So in some parts of Chile, a lot of data could be taken on paper and then passed on to an Excel to see if they use the same metrics. But it is something that is complex. But what is curious is that it is an institutional decision. It is not a problem of resources, or they're just conversations between action areas or legal departments, but data protection and things like that. Hello, very interesting, having to do with food safety. I did not hear. What do you have regarding those foods that are genetically manipulated? That they are putting laws regarding this, because somehow these foods, these transgenic, are also here, not only the pesticides. If I understood the question, is what happens with food safety and the food that is genetically modified? What regulation do they have? Because that is also going to affect food safety, especially transgenic. All of the food that is available on the market is because they have been authorized, reviewed, and there is a procedure that is specific to evaluate food safety, even though they've been modified in a genetic way. There is no food that now is on the market that has not gone through all the different checks having to do with the organisms of those that are genetically modified. There has been controversy over time, but what we have seen over time, and I'm talking only food safety, I'm not talking about environmental impact, what we have seen is that there is no evidence that can show any risk in the consumption of genetically modified food. If there is evidence that comes up, the science is going to say there is an evidence that says the opposite, and then we are going to think about it again. But everything that has been carried out up till now, that are pretty deep and well thought out, it is not something that is done quickly, does not show any risk. Hello, good morning, good afternoon. Thank you for your presentation. I'm from the University of Santiago. My question is a little bit having to do with a challenge coming up later on to see not only medications but also in veterinarians as well as human use in the production of food, mainly because we talk about medication contamination or pollution in the environment, so that'll affect food. The medication also impose pollution in the floors, in the water systems, and we are very connected to the environment. Well, there we are having an innovation in this policy that has to do with the concept of health. We have this in the policy. It is difficult, and we are preparing a publication from Achipia to see how we can bring the focus to health from the national control, food control, but we talk about health, we can see a lot of descriptions in health, but to carry them out is very complex. But it is a first step to recognize that everything is related. And the policy that is going to have an action plan, where the action plan is going to be defined with indicators, people who are responsible, time and resources, that is where we are going to have something that is more concrete whenever possible. And we hope to be able to contribute with this document that we are preparing, that we hope will come out in the second semester. Thank you very much.
Good afternoon, Rodolfo from the Public Health Institute, Statistics Department. Everything you mentioned about AI and how to promote professionals that work on this area on statistics and also that use the modeling to apply it in food safety. Is there any plan to include professionals that aren't experts in the area that can contribute to the research? Not that I know of. I imagine that, well, in Atipia, we are a few, just a few, and we don't have the possibility of hiring someone. And in the future, I'd like to add that type of talent. And I think that Minsal, Cernapesca, and the ASP is also thinking about it. But as a master plan, no, there's nothing like that. I think that due to the progress of AI and that which is so fast, and we cannot react to it in time. So yeah, there is a national policy of AI. We have a policy and an action plan, and there are some guidelines, macro guidelines. But if you start reading about them, we really don't understand what the scope of it, how is it going to affect us, what are the decisions that it's currently making, and that's a little bit complex. Thank you for your presentation, Diego. This is a gift for you. Thank you very much, Diego. Now it's time for the presentation of the impact of climate change in the health of indigenous people, presented by Dr. Carlos Rios Gonzalez from the National Health Institute of Paraguay. He's a medical surgeon focused on control of infections and hospital epidemiology, clinical sexology, health research, aesthetic medicine, masters in higher education of medical sciences, and governability and public policies, Doctor of Public Health, and also in education. We welcome you, Dr. Carlos Rios.
Bueno, muy buenas tardes, casi ya. Para mí es un
Good
placer estar con
afternoon.
usted en esta ocasión
It is a pleasure
para
for
conversar
me
un
to be
poquito
here, and
sobre este tema que para
I'm
nosotros
happy to
en
talk about
Paraguay es
this
sumamente
topic that is
importante,
so
que son
important
las
to us
poblaciones
in
indígenas.
Paraguay
Se
with the
preguntarán, ¿qué
indigenous
hace un
people.
sexólogo
And you may ask
hablando
yourself, why is a
de cambio
sexologist
climático?
talking about
Bueno,
climate change?
básicamente yo
Well,
trabajo más en el área de la salud
I work
pública,
in public health
con énfasis en poblaciones
with an
indígenas.
approach in vulnerable population. And as you know,
Idioma
in
oficial
Paraguay, our
es el
official language is
guaraní,
the
que es
Guarani,
un idioma
which is an
indígena local.
indigenous language.
Todo lo que es
And
la salud
so
indígena, todo lo que es
to
la
us, everything
incorporación
about
de las poblaciones indígenas
indigenous communities
en lo
and
que es el
including
Sistema
indigenous
Nacional
communities
de
in
Salud
our national
es
health system
sumamente
is
importante.
highly
Entonces, en ese
important.
contexto,
And so in that
quiero
context,
contarles
I'd
una
like to share
experiencia
with
que me
you an
tocó en una
experience
revisión respecto a
and
cómo
to do an
impacta el
overview of
cambio climático
how
en lo que
climate
sería la salud
change
de estas
impacts the
poblaciones.
health of this
Casi todas las
population.
comunidades
Almost
de
all of the
América,
communities of the
de
Americas, from
Argentina, México, Estados
Argentina
Unidos,
to
tienen
Mexico
poblaciones
to the United
indígenas.
States,
Entonces,
have
de una u otra
indigenous
forma,
communities, and
esta dinámica, este
this
cambio
af-
ecológico,
this
muchas veces
dynamic change, ecologic
impacta
change,
de
affects and
forma
impacts
desfavorable en la
um,
salud de estas
negatively the
poblaciones.
health of
Y
this
como mencionó
population
acá
of the indigenous
la doctora
people. And
anteriormente,
like the doctor
se encuentran
mentioned
dentro de una
earlier
población
today,
vulnerable.
these
¿Por qué
are a
esto? Porque muchos de
vulnerable
ellos, sé que en
population.
muchos países
And
pasa lo
I
mismo
know
en
this happens in
Paraguay,
many
estas
countries, just
poblaciones
like in
siguen
Paraguay, but
viviendo en
this
áreas
population
rurales, en
continues
áreas
to live in
apartadas,
rural
inclusive
areas,
todavía poblaciones indígenas o familias
faraway
lingüísticas
areas, and
que
there are
viven
still families
aislados del
that are
mundo civil.
living
El
isolated
caso en
from
Paraguay, por ejemplo, le
the
comento
outer world.
rápidamente, nosotros
And
tenemos 5 familias
in
lingüísticas
Paraguay, we have five
que se
linguistic
dividen en
families that
27
are
lenguas,
divided in
de las
27
cuales
languages.
solamente
From
una
those,
es oficial,
only
que es la
one is
lengua
the
guaraní.
official
Entonces,
language,
hay
which is the Guarani language.
otros
So
idiomas
there are
que
many
representan
other
un
languages,
porcentaje muy
but
pequeño
represent
de lo que sería
a
la población
very small
paraguaya.
percentage
Y
of the whole Paraguayan
algo interesante
population.
también es que
And
los
something that's also interesting
indígenas
is that
en Paraguay no
indigenous
se
people in
consideran
Paraguay
paraguayos, se
aren't
consideran
considered,
de acuerdo a
they
su
don't
familia
consider
lingüística,
themselves as
son
Paraguayans. They
guaycurúes,
consider themselves
taba,
Guaycurúes
y un
or
montón de otras
Tawag,
lenguas. Los
according
más
to
conocidos
the
son
language
siempre los
that they speak.
guaraníes.
And the ones
Entonces
that are
ellos se
most known
consideran
are the
como
Guarani,
guaraníes, no
and
paraguayos.
they
Entonces,
consider
aquí
themselves
nosotros para
Guarani as
trabajar
and not
inclusive con
Paraguayan.
ellos
So
es
for us
sumamente
to work
complejo porque
with them in an
ellos dicen,
inclusive
no,
manner
los
is something very
paraguayos.
difficult
No, o
because
sea,
they
ellos
say
son
no,
de
we're
nacionalidad
not
paraguaya,
Paraguayans.
pero
They
los
have that
excluyen, o sea,
nationality,
autoexcluyen y
but
dicen, no,
they
con los paraguayos no
isolate
queremos
themselves.
trabajar.
They
Y algo muy
say that they
interesante
don't want to
también
work
es
with the
que si bien
Paraguayans.
el idioma
Something
oficial
that's
es el
very
guaraní,
interesting is
en
that
muchas
although the
localidades
official language
en
is
Paraguay,
Guarani, in many
los
areas in
indígenas, las
Paraguay,
personas
the
indígenas,
indigenous
no hablan el
people
idioma guaraní.
don't speak
Entonces uno
Guarani,
pregunta, ¿qué
and
idioma hablan?
you may
Hablan
ask yourselves,
alemán
what
y
do
hablan
they
su
speak?
idioma original. ¿Por
German
qué
and
hablan alemán?
their
Porque
original
muchos de los
language. And why
indígenas,
German? Because
por la
of
invasión
the
de las colonias menonitas
invasion of
con
the
alemanes,
colonies of
inclusive hay muchos
Germans
indígenas que no hablan el
that arrive in that area. And there are many indigenous people that don't speak Spanish. So there's a situation that's very interesting that happens there. And I want to show you now a little bit about that. The previous presenters talked about this and mentioned the high impact that it causes in people's health. And repeating this is just mentioning a few of them. And we are very clear about those diseases produced or transmitted by vectors such as arbovirus. And Paraguay had the worst epidemic of dengue with a mortality rate that was very high. And yes, of course, this population that was farther away also were affected by this. And why? Because many of these indigenous people come to the cities to sell their crafts, and a lot of the things that they sell are these brooms made out of juju material that are used in the backyards, and it's very typical thing from our our country, and it is a weed broom, basically, the name, if translated into Spanish or English, because my mother tongue is Guarani, and when I later went to the university and to the city, I started speaking more Spanish. So, what's the dengue dynamic, and why did it affect indigenous people so much? Well, because they were going to the cities to do this exchange, and when they were there, many of them, specifically women, contracted dengue, and they would go to the communities, and all of the community was infected, and some were excluded even or are very far from the family care units and hospitals, and that would affect all of them. And having access to it was very difficult. So, which is why the impact of the arboviruses and the dynamic of the city and taking dengue to very rural areas or taking care of that, it's something that's very interesting and necessary for us. And most of the water consumption there is from the creeks. Do you know what the aljibes are, or also pits, the cisterns, right? Aljibes is the cisterns. So cisterns are one of the tools, but the water isn't treated as in other places. It's direct consumption, and the water that we consume in Paraguay is almost all of it from the well to the mouth. So it isn't common to treat the water. In cities, yes, they do a water treatment, but it isn't as common even in the areas where I live. I live in the outskirts of Asunción, an area called San José de los Campos Limpios de Altapúa is the name of the city. And in that area, we have these water cisterns where it isn't treated at the well. It comes up to the tank, and we consume it as like that. And we're very used to that. However, here a lot of chlorine, and the water is highly treated. So the water that indigenous consume have much more contamination. And something that is very interesting as well is that respiratory diseases that I did not include in this presentation are genetic diseases. And I was talking about the Menonitas. And what they do is they provide with work to the Mennonites, and they irrigate with agrotoxical elements with these airplanes where they use these chemicals, and there are a lot of children with affectations due to these pesticides. And this has a high impact in human health, specifically due to the movement of the communities where we're in, that were basically self-sustainable, and they had to move to certain areas or to the capital or to the cities to try to find different ways of life that weren't the traditional ones, and that causes a lot of issues. And like my colleagues mentioned earlier, relating this to food safety with the previous presentation of the doctor, this is something that's very concerning because in the past, Guarani people were nomads, and and they were collectors. And compared to today, and to other indigenous communities, what they would do was that they would go to a place, they would uh, get everything they could, they would move to another areas where there were more things, and then they would go back to these areas. And this is something that they cannot do today. Today, they cannot do that, and they are farther away. They cannot do these things that they used to in the past. And there's a saying in Paraguay that says that the Guarani people eat everything that they have when they have it. So if you give them 10 kilos of rice, the rice would run out that very same day. That's a very common behavior of Guaranis, because they didn't have the culture of saving, and that is why we did this. They got along with the Spanish very well, because the Guaraníes had the capacity of adapting very well to any system, and the mix of indigenous with Spanish happened like that. A lot of these mix of races, um, there was a lot of mix of the races in Paraguay. There wasn't a resistance. It happened right away in Paraguay, and it happened a lot. And something else that's very interesting as well is that until 2010 approximately, and although since the 82 the indigenous peoples were recognized and with the official language, until 2010 there was no law for the indigenous people, and they were highly excluded, and the health system as well, where the health professionals didn't want to work directly with this population, and due to stigma and self-exclusion is something that's very typical as well and frequent in this population. So you they don't go to the primary care facilities because they are going to look at them differently with disgust. They will treat them differently. And even a few years ago, in the 90s approximately, many professionals decided not to talk in Guarani because it would cause discrimination. During the dictatorship, it wasn't something that people wanted to do, and wanted they didn't want to talk in Guarani because it caused the stigma. And after the law, the indigenous population and everybody should speak Guarani language, and specifically those who work in rural areas. And this is something classic. With the mom, you speak Guarani. With the parents, you do speak Guarani as well. And even uh, we speak in Guarani because that's a traditional way we speak and communicate with each other. Of course, all of traditional practices have been highly affected. The ceremonies, the funerals had a huge impact because Guaranis and other indigenous communities were buried in fetal position in urns, and that's something that urns like pots, and they couldn't do it anymore, and that caused a lot of impact, and it was badly seen for a very long time for them to do these practices. And all of that created a cultural shift, and even labor um, for women. They would do it with dried leaves, birthing. It was a way they would remove mosquitoes by burning some weeds, and all of those things that were part of the identity change. And that's basically due to the movement of indigenous people. And you might think, why is that related to climate change? Well, it's because we took away from them areas where they used to reside, and we took them to the city. They cannot practice their culture as they used to, and that affects mental health. And with these few minutes that I have left, I want to share with you some studies about research in indigenous communities and in our in our health institute of Paraguay, and in the area that I preside, we work a lot on qualitative investigation research. Through the morning, I saw that most of the research are quantitative, and in turn, we promote qualitative research because our main focus is indigenous people and vulnerable communities and groups. And we work from up close with ethnographic research, phenomenological research, and even we finished not so long ago a research of a participatory action with indigenous communities where we addressed resiliency and adaptability to climate change in certain indigenous populations, which is why I want to share with you the way that we look at reality, and that's how we call it. Qualitative research, it's a quantitative is a way of looking at it, and qualitative is another way of looking the reality. And we are very interested in having the holistic approach focused on the people, which is why we are trying to promote an inclusive action. We have a master's in public health in our institute, and one of the major investigation lines is to do research in vulnerable groups, and we put the emphasis on qualitative and mixed research, and we try to address everything that's related to all of those different approaches. And I shared this with you, and this is something that we did in Brazil in the Tuxa and Pancararu communities. And we saw that the traditional way of living was affected, the culture, the, the cultivation, and they had to use aerotoxins because what they used to do in the past was that they would prepare drinks that were in the past used as pesticides. And same for Bolivia. We have a close connection with them, and they use insecticide as well. And we in Bolivia we see a lot of child malnutrition, and that's related to the reduction of foods that they would consume. And in Paraguay, I'm showing you this directly. And in Paraguay, we traditionally consume 2 to 3 types of meats: cow meat, pig, and chicken. Fish, little to none, and basically that. And chicken, a lot of chicken because it's the cheapest, it's what we consume the most. And we had a research in indigenous population, Guarani people, where we saw that they would consume about 10 to 12 types of meat because most of them had wild boars, deers, snakes, and all of those animals are eaten. So there is a variability, and if moved to the city, when they moved to the city, they couldn't do it anymore because it's something that Paraguayans think it's not right to eat snakes, right? And they stopped doing it. And one of the research that we did was exactly that. We saw the perception and adaptability of climate change, and we used a methodology that's called participative action. And the first thing that we did was do a diagnosis of the situation and how the perception, the thinking of the Guarani indigenous people was. And this is something that we did in the Guarani language, very close. It was very interesting, actually, for us who work in sexual health. We want to have peers and to work with indigenous people. We work with them, we train them, and they become experts in sampling collections, etc. And we do all of this with interviews, with recordings, which is very interesting. And we do the diagnosis. We diagnose what are the main issues related to climate change, and then we do a meeting, and they call it Atewasu. This is a community meeting where we have these huge pots of mud where they do prepare the food. Everybody eats there, and during this meeting, what we do is try to check, I mean, talk about the adaptive strategies that they are proposing in order to improve.
They say we cannot cultivate this because there is too much of this or too much of that, or basically with them we could identify what could be done in the community. And this is a community that is surrounded by 5 kilometers of forest. They're in the middle of the forest, surrounded around 5 kilometers around them, so that it's not easy to come in, so to say. So they try to conserve part of their culture, and we try to work with them. So that is what we found. And of course, they also see a lot of impact. Oh, also here what I was showing, and I was trying to say these are real pictures that we took. You can see the characteristics that they have, and this is because there is a mix with the Mennonites as well. And one of the girls was albino. So this is part of the symbiosis and the capability of being united to different cultures. So they are very characterized by these things, and that is very interesting that I wanted to show. So that would be it then. So what are the challenges for research to work in these methodologies that are participative and to try to promote, and this is something that I want to repeat, some try to work with different visions to be able to come closer to these communities, to be able to get to know the knowledge from before, the ancestral knowledge, to see how they deal with climate change and to see how they cope with all of the adaptation that they need to be able to continue. So this is very important, even for us, to see the ethical focus. It can be very complex. So that would be it for now. I'm sorry I passed my time, and I would like to thank you very much for this space, and I will be open to any question you might have. Thank you very much, Carlos. We are going to leave all of the questions for your lunch. So if you want to come close to him, feel free to go up to him and ask him your questions directly. And we will give you a small acknowledgement from our institution. So now we are going to begin with the different papers that are going to be presented here in the institution after the evaluation. We have selected 3 of these, from which the first one is going to be to Mauricio Araya from the Chemical Environmental of the Health Institute. It's the evaluation of water in rural areas of the metropolitan area. Mauricio? We do not have the presentation yet, so we're going to change real quick. And we are going to have Janina Corrotea Varas from Food Chemistry of the ISP, and her work is called Evaluation of the Compliance of uh, Labeling of Aspartame in Children's Products of National Consumption. They're loading the presentation. That's why we have a short pause. We are waiting just 1 second because they are loading the presentation. 1 second, please. So, good afternoon. I'm going to present a study that was carried out in the chemical part of the food in the department, National Department in reference of environmental health, and it has to do with the evaluation of the fulfillment or compliance of labeling of spare time in the children's products that are consumed nationally. First of all, as introduction, I would like to explain what aspartame is. Aspartame is a artificial sweetener that is used as a substitute for sugar for the reduction of calories. This aspartame is considered an additive, and for it to be considered a substance as an additive, it has to fulfill a series of requisites, so to say, among them something that is very important, which is toxicological report as well. And this is going to show that the additive is safe for the consumption of the population, and it also has to fulfill a technological role within the food. And the technological role in spare time is sweetener. In this toxicological evaluation, we have a spare time, and it tells us how much can be eaten per day, the ingestion per day. And this is the amount how can be eaten during every day for the rest of their life without a risk for their health. And for spare time, it's 40 milligrams per kilo of body weight. That is due to the person's body weight. And that is very important that we keep that in mind. And to be able to ensure that number, you have the maximum limits permitted, and that is the milligrams of aspartame per kilo of food. So it's the highest concentration that can be added of this additive to food. This does not mean that this is the dose that has to be added. It is telling us that it is the maximum concentration it can have to ensure that consuming this product is safe. And where can we find a spare time? We can find it in different food categories. For example, juice, drinks, dairy products, cereals. But we're going to focus on certain matrices for this study. And nationally, who regulates the additives is the national regulation, and you can have in this national regulation document that it is allowed to use a spare time. However, it is used only with uh, the limited amount So if we go to the international guidelines, which is the Codex Alimentarius, which they have mentioned before. They talked about this codex, which is a series of norms established by FAO and WHO, and its objective is to protect the health of its consumers and to establish practice of fair commerce or fair business. And they have different norms and a consensus in which Chile is also participating. And a spare time is permitted and allowed by category of food and as well as being regulated with the limited of maximum permitted. But this has been we have been working on updating this so that the guidelines here can be much more rigorous, and soon it is going to be updated to codex. So the additives that currently do not have a maximum amount permitted will have it, and they would be in the category of food, which is a progress that would be very good, positive for our guidelines. So what is the problem that then started that triggered for us to start the study? Is that WHO in 2023 carried out a declaration from some of the studies from the cancer studies, saying that spare time might be cancerous or produce cancer as 2B. And here we think that this news could generate alarm in the population, so we wanted to then carry out a study. This is the first part of the study that was carried out, and in this first part of the study, what we carried out was an evaluation of the labeling of the nutrition regarding aspartame. So we corroborated that what the label says and declares is actually what the food contains. And we compared that with the maximum limit that is permitted, and we went to the international guidelines since our regulation does not have it. And our main objective is children, because if we remember the children, which is a measurement for children, it has to do with their weight. Therefore, they are the ones who are most likely to go over the safety dose due to that they are smaller and they are the higher risk. Having said this, the objective is to analyze the concentration of aspartame in children consumption of foods, for example, drinks, nectars, juices, yogurts, different milks, and verify that the information declared on the label is what is found in analysis. As a method, the sample we had first exploration in seven communes, which are Ñuñoa, Peñalolenda, Reina, Curacaví in the metropolitan region. The total number of samples corresponds to 85 products that declare that they have uh, these sweeteners. And in food, we already described them in the objectives. We have these different drinks, and they are all in individual formats centered on the children's population. And from these samples, for each kind of category, we sampled two available brands when we purchased, and each one of them, we selected two different lots or batches, and each one had 6 units to be able to sample. So regarding the method for analysis, we have to we take 10 ml of homogeneous sample. We add water and some reactivating so we can see the different protein and different fat, and then we have a process to filter to have our extract cleaner, and then we filter with a smaller porous to be able to go to detection with HPLC DAD. So regarding the results that we obtained are presented segregated by category according to codex. And in this case, we have the drinks, and we can see these are fantasy drinks. And we can see that there are some drinks that do not declare they use aspartame. So you have the red and yellow, whether they declare they have aspartame or not. We wanted to make sure the label was correct, and we can see that We analyzed these samples that did not have aspartame, and they are under the limit of quantification of our methodology, therefore they are according to the label. And regarding to the samples that did declare having aspartame, we can see that which one was declared is in blue, and what is analyzed is in orange. And we can see that in none of the cases what we analyzed was above what was labeled. Therefore, there is agreement with what the label says, with what we found in the analysis. There is also something very interesting in the fantasy drinks, because you remember that the sanitary guidelines does not have a maximum limit, so we use the codex limits. So there are two that go over the limit, which is the orange fantasy, but it does comply with national allowance because it does not have a limit. But since it will be soon updated, we would then have these matrix that would not be fulfilling what would be the new guideline. Therefore, this industry would have to reformulate their products so that they could be within the allowed ranges. Regarding the nectar drinks, we analyzed 18 samples of these. Some of them declared to not have any sweetener or aspartame, I mean. And we found that all the samples were under the limit of quantification. Therefore, this was also very gratifying to find that the industry, that what they declare on the label is actually what we are finding in the analysis, and they are fulfilling the guidelines as they are at this point. To be able to see the codex maximum limit permitted, we can see that these different flavors, does not reach the maximum permitted, therefore they are in compliance. We also evaluated powder juices, and you would think, why would you think about powder juices for younger populations? But there is a part of the population that does have powder juices made out of powder, to not spend as much money. And many times the family prepares these juices and give it to their children as a snack, and the drinks as well. Many times the drinks for smaller children, we should not be giving them anything under 2 year old, but culturally they sometimes does happen. So the powdered juices were also evaluated because it's a reality that children do consume powdered drinks. The family prepares it for them. So we saw the different flavors, and we also found that all of these do have non-nutritious sweetener, and they had aspartame, and we could see that there was a agreement, or it matched according to what was in the label and what we found. In yogurt, they did not declare using aspartame, and we found them all under the level of quantification, therefore they are correct. And in milk, we have the same situation. They do not declare they have it, and the 18 samples that we analyzed are all under the quantification limit of our methodology. And now to the conclusion. I'm sorry, I'm getting out of time. And regarding the conclusions, having to do with the analytical study, we confirmed that all the categories of food that were analyzed that did not declare aspartame on their label, the laboratory did not detect this sweetener. In other words, the result is lower than the limit of quantification. All the samples that do declare aspartame, the laboratory did detect the presence of this sweetener, and we could see that there was concordance between the values that were declared of aspartame and those that were determined by the laboratory. From the 85 samples that were evaluated, we can observe that the manufacturers adjust to the local norms or guidelines, and they are indicating it in their labels. And regarding International Codex Alimentarius, some of the samples of fantasy drinks, as we could see, go over the maximum allowed limit. So those were the only ones that would not be within those limits. And before I thank you, this study does not finalize here, because this is just something that we are using for the evaluation to see if these matrix with the concentrations that were determined analytically can go above the IDA, and that is in a poster, which is I would like to invite you to see because the finalization of the study that we carried out to see if there was any impact or not. And that is poster 107, and I would like to invite you to do that because the evaluation of the because it is very important to see if the children are eating more than what they should be eating according to the dose and their weight. Is there any question for Janina? Yes, there is a question. I would like to congratulate you. It is very interesting to see your contributions. Sometimes we don't take what does catch my attention. What would be the explanation of the bias and the sensitivity where you detect under the quantities that they declare that they have placed? What is the methodology strategy that you are using, and why did we not see the same sensitivity of the concentration that should exist? Applying the uncertainty of the methodology, it goes very close to the value that has been declared. However, the value of the concentration is being analyzed, but if we apply the uncertainty of the methodology, we come closer to the value. I do not know what methodology the industry uses to be able to determine the concentration of this sweetener. However, I do believe that our determination has been very strict. It has good recuperation, and the most important thing is that we found all values that are very similar or under the declared value. Therefore, that is something that gives us hope because if it were over, then we would not be fulfilling with the industry with, according to what has been analyzed. So we are also implementing this methodology through HPLC mass mass, which is much more specific, much more robust as well in this methodology. But even so, I do think that applying uncertainty of the methodology, we are close to the value declared. It is not a problem for us, nor for the evaluation in general. So we would like to give an applause for Janina. Thank you for your presentation, and the same. I think the visit to the poster is very productive. So now we continue with the presentation of Rayana Palairini from the Department of Risk Prevention of the Environment of the University of Tecnológica Metropolitana de Santiago with the application of teledetection for prediction of infectious diseases in water in Chile. So, Rayana, go ahead.
Hello, everyone. Good afternoon. I came here to speak with you and tell you about the challenges and the potential about remote sensing. Remote sensing allows us to look at information but not measure it, estimate it from afar. So the idea for today is to talk about this. But before I do so, I'd like to introduce myself because I am not from the health uh, sector. I am from meteorologies and I am from Brazil. Yeah, I think you can tell because of my accent. And I have been here in Chile for 4 or 5 years. I studied meteorology since my graduate and my undergraduate, and I did my thesis on that, and I came to Chile to do a postdoc doc studies in Catholic University. And the idea was to do the relationship and to relate the health public policies. And when I did that, I was very surprised because I was always um, focused on being a meteorologist and the changes, whether it was going to rain or not, what will happen with the temperature of the day. So getting into this area of the health area, it's something that's very interesting for me. I want to share with you today a seed fund that I received of a project that I participated in, funded by the Inter-American Institute of Climate Change, and we participated in a workshop, and the idea of the project was finding researchers of different areas: climate, health, environment, people who were from the academia, participated in the academia, were decision makers, people from civil society, and the idea was to integrate the knowledge of all of them. We participated in this event, and we had to solve a question of, of this research of this course. And I was thinking, what can my question be if I put together all these three areas? And like I mentioned here today, the WHO has a phrase that's very interesting that says that we're going to have about 250,000 deaths or more due to climate change. And I think that to me is something that's quite severe. And all of the management of the climate and extreme climate, extreme temperatures, the increase of the water level, the increase of CO2, all of that affects the health of people that have different diseases. But the research on climate and health isn't something new. And that's something that made me surprised because uh, I decided to use remote sensing to do so, and using satellite data to do this isn't new. There are some works from the 2000s that show the relationship of climate variables with cholera, diarrhea disease. So, with this group, we thought, okay, maybe we can propose, if possible, to do a monitoring system of low cost by using satellite data to prevent or minimize contagion of infectious diseases transmitted through water. So we started with that question. We started this pilot project in the metropolitan region in Chile, considering a region that has more than 7,000 million people, and a metropolitan region that has climate characteristics that are interesting, and because they are close mountain chain on one side and on the other side the sea, and that means that this is an area with climate conditions that are very specific, different than other areas of the region, such as Brazil, for example. And after looking at the reports of the ISP, we saw that Salmonella was a pathogen that had the most amount of biooccurrences or confirmed strains, detected strains, and we thought about that. We started from that statement. Then, after talking with the ICO professor and Andrea Moreno, they said, okay, Raina, we have a database because we did a study doing on 2019 from 2019 to 2023 where we collected samples of those physical chemical parameters on the Maipo River and Mapocho River. We processed those informations, and we have identified whether we have identified salmonella or not in certain areas of the Mapocho and Maipo rivers. That's work that's already published. And I said, okay, yes, I think that we can use that and relate that to satellite data. And that was the first idea that we had to do the identification of satellite image of high resolution after understanding what's the reflectance of the images through the spectral images on water courses, and try to find if there is a relationship between the environmental parameters and reflectance, considering this figure that we have down here where we have these different images And we can identify whether we have vegetation, water, and we can verify and check that through satellite images. So we continue with this methodological process of acquisition of acquiring data, the use of images with a predictive model, then we use an interpolation model, and then we discuss what we could find. And what is a possible relationship between the pathogens and the situations and conditions around the rivers? First, we found that the one of the indexes, which is one of the normalized index of water that uses a width of it's seen as green and infrared. And with those indexes, we were able to identify if we had a body of water or if we had any other kind of surface. And we consider that, yeah, we didn't have a cloud coverage that is we shouldn't have one that cloud coverage that is more of 50% with the atmospheric conditions. And we also looked at the coverage of the land use. And we saw that in figure A, we had dots in red, the dots that we sampled, and in yellow, you see the areas that are agricultural areas. And we also have urban areas. And these rivers and around the rivers, you find two types of land use. After that, we used a predictive model where you can see the databases and 01, and we use those samples with an extra 3 classifier model, which is similar to Honda Forest, and that uses a sample randomly and tries to do a prediction and to understand what's the most likely area of occurrence of those pathogens. And we use this interpolation system uh, with the distance, and we found three main points, which is the risk area with the high probability of having those cases affecting the people with these salmonella bacteria. And we had a 59.2% accuracy, and we know that this isn't the best accuracy, and we need to work on that a little bit more to have better values. And we can already see an expectation and a potential of how to use or on using these type of techniques and of the analysis of the area of health. And that was the goal, because this is a pilot study, an exploratory study, to try to understand if it was possible to find a relationship. With that in mind, we started thinking about the social aspects. Who are the people who are affected by this? And we looked at the social demography data from Chile, and we also looked at the regions where there are shanty towns, informal settlements. For example, we found in the map up here, where you see the purple areas, those are shanty towns in Puente Alto. And over there, we have about 11 shanty towns with more than 4,000 families that are living on the edge of the river that are directly affected by it. So part of our team did a visit to the area and got to see the reality of the people who live in this area. And we had to do science, but also include the transdisciplinary area and see how to apply that in the society and how we can help the society. So, some final comments. We can answer that first question. Yes, it is possible to do it, but we still need a lot more to do in order to have a an alert system, low cost alert system, because we need to have more data, more samples. We need collaboration between the different ministries, because sometimes, as a foreigner and as a scientist of climate, I see that sometimes we need to have more of these conversations between the Public Health Institute, the Ministry of Health, the Ministry of Environment. And I was delighted with the lecture of Sandra, because they are trying to do that from a point of view of the public policy. And it's important for the different institutions to start speaking the same language and talking to each other and looking at the same and working for the same goal, not doing things isolated. And so from the academia, I see that there's a need of having health data that's open, data that was already collected, and not as it happens today with the ISP report, where you can only see a, a group of information of the annual amount, the annual data or the monthly data. But as a scientist, that's not enough for me, because I need the daily data in order to do a comparison with the satellite data. So I think that we still need to have this view of the Society of Public Policies and open data uh, platform, where we can all talk to each other from our different sectors, because in the end that will allow us to grow and to develop better work and to have better progress. This is a team that worked in this project and helped me make it happen. I'd like to thank them, and also thank all of you for listening to my presentation. A very small question, please. If not, you can ask her during lunch. All right, thank you, Rayana, for your presentation. I know we are behind in schedule, at least 30 minutes with lunchtime, but I think science should be put first, and we can wait a little longer for lunch. We have the last presentation now from Mauricio. Mauricio, please come to the stage to talk about untreated water served in the metropolitan region in Chile.
Thank you very much. I would like to thank the organizing committee for selecting this work because it frames a work together with others, and I would like to highlight what the previous person said. This is interinstitutional work, and now I am going to give you more information. This has evaluation of the quality of untreated or raw water in the rural sanitary services in the metropolitan region. So, a quick summary to be able to bring up the information, the most important information in 2024 or last year, we carried out as Public Health Institute together with the Seremi of the metropolitan region, we carried out a study focused on evaluating the quality of water that is used in rural sanitary or health services. The ISP is focused on the analysis of raw water or untreated water. And in parallel, the Seremi in their ministry program are talking about the quality of drinking water. We had 87 wells that were tested in the metropolitan region, and as a summary, 97% of the samples complied with the microbiological parameters, and 31% of the samples presented numbers that were anomalous, or they were over the physical chemical numbers. The positive things from this work is that we were also able to evaluate this in the sector or focus it, identify it in the south, in the southern side of the city. The drinking water, rural drinking water services are services that deliver drinking water to rural areas that are uh, brought together by committees or through cooperations with the objective of being able to deliver drinking water of high quality and in the amount that is required. These rural sanitary services many times present problems related to a limited infrastructure, availability of technical and financial resources, and availability of the water that is available. To be able to assess the quality of the water, there is a legal norm, Supreme Decree 735, that focuses on the microbiological, physiochemical parameters. There are some external pressures, like, for example, agriculture, the growth of the population, and the climate change that also affects the quality of water. As objective and methodology, our main objective of this is to be able to evaluate the microbiological and physical chemical water quality that is used in the rural areas. As I said before, there, for the development of this work, we carried out an interinstitutional collaboration. The collection of samples was carried out between April of last year until November of last year, and it was carried out in 18 different campaigns to be able to have samples, collecting 87 samples of untreated water. The analysis is focused on the microbiological and physical chemical parameters, which you can see on the screen now. These are the points from the samples in the metropolitan region, outside of the metropolitan or urban area. It was 87 points of samples. We had a coverage of 77.7% of the total of the sanitary services that are available right now in the metropolitan region. The 100 in total services that exist serve 200,000 people in the metropolitan region. The analysis methods that were used, we were able to put them in four large groups. The microbiological parameters, specifically for the evaluation of feces, and also metals that were metals and metalloids that were the classical chemistry parameters, ammonium, pH, etc., and also the anions that were evaluated through ionic chromatography. Some of the results, microbiological results, you can see that 96.6% of the samples complied. 3.4 were not did not comply, and these were three different wells that were affected. And as you will see in the presentation of the poster that is being presented now, these samples correspond to a sample that was carried out post high rain moments. However, these three samples that were evaluated regarding what was carried out by the Seremi for drinking water, they did not identify these components, only in untreated water. And with the physical chemical results, you can see on screen the distribution of the different results for the 20 analyses that were carried out for the samples. You can also observe that there are 27 points that exceeded at least one parameter within the norm. The main incidence was in the presence of nitrate, with 21 samples over the normative parameters, sulfate, and total dissolved solids. And regarding these three main incidences, we can see the nitrate, nitrate, and sulfate and their georeference. And in the nitrate, you can see that there are 20 points that went over the maximum, which is 50 ml per liter. And they are all focused in the south of the city. And you can also see in the image the diagram, a heat diagram, where the values of higher concentration are in red. And regarding nitrite, there are 6 points that had values above what was regulated, which is 3 milligrams per liter. And they also the identification and finally also sulfate, where there were 5 points. The maximum is 500 milligrams per liter. And there they have defined patterns in geography. Now that we have the results in untreated water, we then have to evaluate the drinking water process comparing the results from the Seremi. And we compared these samples of untreated versus treated water, which were tested in parallel. And you could see that the treatment, to be able to make it treating water, allowed to reduce the concentration of many parameters. However, there is a persistence of in some rural areas in which there are still elevated levels of nitrate, even after they went through the process. Therefore, as a discussion of the results, we can focus on 4 main points regarding microbiological safety. Most of them samples presented no pollution, which means that there is safety or security in the sources. Regarding physiochemical parameters, 31% of the samples went above the normative limits, at least in one parameter, especially for nitrate, nitrate, and sulfate SDT. The hotspots that were identified allowed us to evaluate the concentration of those who would exceed and the areas, especially in Padre Hurtado, Peñaflor, Melipilla, Paine, Talagante, and Curacaví. Regarding the efficacy of the treatment of drinking water, the difference between untreated and treated water, there is good elimination of metals as well as the persistence of nitrates in post-treatment and some post-treatment points. And as a conclusion and recommendation, we can define 4 points. The study shows the need to strengthen monitoring, treatment, and water management in the rural sanitary services. And it's also key to place special attention in those areas that are identified as critical. There are preventive and corrective measurements required to be able to ensure the fulfillment of quality standards. And these actions are key to protect the rural communities. And finally, as final summary, it is very important to highlight that this geographical focus that was carried out, there was an interinstitutional collaboration and optimization of the treatment and um, the treating of the water, which is very important. Thank you very much. Thank you very much, Mauricio. One question. There is space for one. No, you can discuss it over lunch. Great. Mauricio, thank you once again for your presentation. So now there is a recess to finally go have lunch with a lot more energy. It's going to be extra tasty, and we will be here at 2:25. We will be 2:45. I'm sorry, we will be a little bit delayed, but we will be back in 45 minutes. So don't forget to look at the posters. We will see you in a little bit later.
estén alrededor, que por favor pasen a el módulo central de nuestra carpa plegable. El término correcto. Ya. Entonces, señoras y señores, muy buenas tardes. El Instituto Salud Pública de Chile les da una cordial bienvenida y continuación a
We
repetir la
welcome you
mañana y
and let's
agradecemos su
continue
presencia en la
today's
17ª versión
program.
de las
Thank you for
jornadas
being here
científicas.
on this
Este año nuestro lema también
scientific
cabe
conference.
recordar que es
It is
el
important
ISP,
to mention
impulsor de
that the
ciencia y
Public Health
regulación para
Institute
la
is a
salud
promoter of
pública.
science and regulation
El
for
eje
public
temático
health.
correspondiente a este bloque se
The topic
denomina
of
el
this
tercer foro
blog
del
is
Instituto
called the
Nacional de
Third
Salud
Forum of
a cargo del Departamento Nacional de
Health
Referencia en Salud
Conference
Ambiental.
in
La primera
environmental
conferencia
health.
que tenemos
The
en
first
este
conference that
2024
we
del
have is the
climático
impact of
en la
climate
salud de la
change
población en
in
Bolivia.
on the
Su
health of indigenous
expositora,
communities.
señora
The
Fabiola
presenters
Vidal,
Fabiola Vidal,
bioinformación,
biochemist,
magíster en epidemiología,
masters in
magíster en
epidemiology,
bromatología,
master in
especialista en
bromatology,
vigilancia
specialist
epidemiológica
in
y en desarrollo, innovación
epidemiological
del
surveillance
sistema de gestión de la
and
salud por
development
ensayo y
of
calibración según la norma ISO
calibration
17025.
according to ISO
25 años de
1725,
trabajo en el Grupo
25 years
Nacional de
of work
Laboratorio de Salud y LASA
in
en el
the
Laboratorio
lab
de Control de
of health,
Alimentos, Laboratorio
LASA,
de Salud
control of
Ambiental,
food, environmental
coordinadora de la división del control de
health,
INLASA,
and
coordinadora de la red de
coordinator
laboratorios oficiales
of
del
the official
análisis
labs
de alimentos de
of
Bolivia,
analysis of food in
coordinadora del Grupo
Bolivia,
Técnico de
coordinator of the
Calidad de la
technical
Red
group of the
Interamericana de Laboratorios de
Inter-American
Análisis de
Network
Alimentos,
of Food and of
dejo con
Analysis,
ustedes a Fabiola
RILA.
Vidal.
Fabiola Vidal, you have the floor. Thank you, everyone. Thank you to the Public Health Institute of Chile for allowing us to be here and to be part of this conference and the Forum of Climate Change and Health of Bolivia. Thank you very much and good afternoon, everyone. I'll show you now my presentation of the current situation of Bolivia in terms of climate change and all of the effects we are experiencing in these last years due to these characteristic that we have of the climate and the problems associated to climate change. And the most important ones are natural disasters such as drought, flooding. Those are the effects with the most frequency and the biggest impact with a population of 11 million amount of inhabitants. That also affects food safety and also water safety. It's been an increase in the amount of forest fires and the propagation of diseases in areas where there were no signs of those types of diseases. As I was saying, flooding is one of the natural disasters that's most frequent in Bolivia in the past year, and in 2024, the rain season that started in November extended to these days. We are still experiencing rain, something that wasn't frequent in our country. March was the end of the rain season, but we are currently experiencing rains, and this is a phenomenon that has happened after 40 years and still affects us. In this season of 2025, we've had 55 people dying that have died, 8 people that have gone missing, and 600,000 families affected by these floods. This is a phenomenon that has affected all of the departments of Bolivia and areas of Bolivia. And in March of this year, Bolivia declared national emergency due to the floods. And as a comparison of what happened in 2024, the amount of families affected were 235,000, and as of 2025, we have increased that number to 600,000 families affected by these disasters. Bolivia has 342 municipalities, and 114 municipalities were affected in 2024, and as of 2025, 211 municipalities have been affected by the fires and by the floods, which is double. Another important event produced by climate change has been the forest fires. Forest fires have increased significantly in these past years, and the most important ones in have occurred in 2024, where we had more than 3,000 spots of fires, and with the air contamination being incompatible with human life. Between May and October 2024, Bolivia faced the worst crisis of forest fires and declared a national disaster, and the Ministry of Health declared it a health emergency. This is the first time that this happened that in Brazil. And the main causes of forest fires have identified several, such as extreme drought due to climate change, agri traditional agricultural practices, such as burning uh, weeds to eliminate what has been left of the agricultural production. They burn in the certain areas, but that has extended because of the droughts. And then we have deforestation as well. More than 10 million hectares of woods have turned to ashes in 2024. Areas in Tiquetania, Amazonia, and the Amazonas and Santa Cruz, which borders with Brazil. What has caused these forest fires? Like I mentioned, health alert was declared due to risk of health, and one of the main risks are respiratory symptoms, cardiovascular symptoms of the medical personnel, firefighters, and also the population that is in contact with the smoke. The smoke has traveled to several municipalities that were uh, that didn't have forest fires, but it has even reached La Paz to the other end of Santa Cruz. There have been acute intoxications due to carbon monoxide, burns, and other topical reactions. This has also extended through the Amazonas and has affected the fauna, the wild animals living in this region. Some other consequences from forest fires are diseases caused by the food. And as I mentioned, the ashes have been so strong and the expansion of the smoke has been so much that there have been contamination of water due to the ashes. In Bolivia, people consume well water, and some regions have been affected by ashes in the water and other chemical substances in the water, and these chemical substances produced due to the fires. In Bolivia, there's also mining operations, and that's also contaminating the rivers and the south basin as well as the north basin. And although this is unrelated to climate change, this uh, exemplifies how we are not taking care of our resource that's so important, like water, that will be so significant in the future. And we are currently having basins contaminated with mercury, and this is because of the gold exploitation. And this is something that's important that our authorities need to consider. Uh, the Ministry of Environment, and they need to consider the health and the water as well. Regarding the droughts, this is affecting directly climate affected due to climate change. There's no possibility of irrigation, and regarding flooding, in the one event that we had in Santa Cruz, which is a livestock region, and because of the drought, of the floodings, they have had to move the livestock, the cows, to other regions and to other places, and they couldn't find a high place to live and to move these livestock. So a lot of animals have died because of this. Same thing happened in the west with the sheep and goats. They have also been affected by that, and there has been a lot of loss in these livestock, which affects the food safety of our country. Another effect of climate change is the spreading of pathologies produced by vectors in areas where they wouldn't um, be seen in the past. We have a study of Oropuche and detection of Culicoides paraensis, which is a vector that transmits Oropuche, and that has been found in the Department of La Paz, the Municipality of La Paz in Uripana, an area where we wouldn't have this disease, but in 2024 we have seen the presence of Oropuche, and it is because of the insect located in this locality in La Paz. Like so, for Ismaniasis visceral, there has been a presence of disease in Villamonte and Tarija municipality, and since 2022 we have been working with those cases, the presence of this disease and Luxioma crucis, which is the vector of this leishmaniasis, another disease that wouldn't be seen in the La Paz virus and the La Paz municipality, and that wasn't frequent in this area, is hantavirus transmitted by rodents, and in 2025 we've had 20 cases of hantavirus and 7 people have died because of this. In the Yungas region, La Paz, which has a tropical warm climate, there was one sick person every 2-3 years, but only as of 2025 we've had now 20 cases with 7 people dead because of this. The institution where I work, which is a National Institute of Health Laboratories, depends on the Ministry of Health and Sport, and we have some projections to support the in research the ministry in order to detect diseases from the genome aspect and bacterial aspects. And among our projections and works that we have done, there is one that has an approach of One Health that allows us to identify pathogens in any kind of sample. We also do genome surveillance in terms of virus and bacteria, and we are making progress in this as well. And we want to be part of this work in the region in terms of research and of climate change. We also do investigation application of biotechnology with an approach on disease treatment to support agriculture, the development uh, of food and improve food, biofuels, care of the environment, recycling, treatment of waste, and the cleaning of contaminated areas due to industrial activities. We're also doing research on analysis of heavy metals in water, food, and human samples of areas where there are basins that have been contaminated by heavy metals. One of the investigations we have been working on very closely with the Institute of Public Health of Ecuador is the implementation of the technique of using a sterile insect applied to the Aedes aegypti mosquito to prevent the transmission of diseases as dengue, chikungunya, to control of population of these mosquitoes and mitigate the risk of these diseases. We do trainings with people in Ecuador, and we have applied this technique through the agency, Bolivian Agency of Nuclear Energy, and we are already at the stage of implementation. So we are halfway through our investigation, our research. This is the work that we have been doing in order to control these diseases. We've had donations of equipment, training to Bolivian personnel in the institute where I work at, and we are in the first stages of implementing this methodology. And now, regarding the institute I work at, we have accreditation ISO 17025 for food, water, and genetically modified organisms. We also have another ISO accreditation 15189 for diagnosis, clinical analysis, accredited also with ISO 17043 for trials. And we also work in public health. We're a network. We are the network of analysis of food, and this network has about 25 labs that work together, and that allows us to control whenever there are emergencies, to take care of food safety, and that's what we prioritize for our country. Thank you very much for giving me the opportunity of being here today. We have time for a question. Anybody would like to ask a question? No question? Yes, well, Fabiola, I would like to know about what would be the main issue you see or do you believe is happening in Bolivia? And from what everything you mentioned to you, what do you think is the most critical issue in Bolivia? I think the most critical issue today in Bolivia is forest fires because it has been often, and we've always had forest fires, but now uh, even more. We've always had in Santa Cruz, La Paz, those areas, but now um, it's we haven't been able to control it, and this is something that's clearly not going to stop because it's related to agriculture and agricultural practices that are uh, used in my country, and that, along with drought, I think it helps me answer these questions and saying that this is the main issue that we have. To destroy everything that we have agricultural production is the easiest thing to do is burn, and then when they do so, it spreads, and with the winds, it spreads to the east. No questions, okay? Thank you, Fabiola, for your presentation. This is a gift from our institution for you. Big round of applause to Fabiola. Thank you for considering me for the picture this time. Now we have a presentation via streaming. The next presentation is called The Development of a Monitoring System of Risk uh, for Forest Fires. Mariana Manteca from the Ministry of Health of Argentina. Mariana is a doctor of biological sciences of the University of Buenos Aires with a career in epidemiology. She is currently director of the National Center of Diagnosis and Research in Endemo Epidemics from the Ministry of Health of Argentina, where she coordinates and develops research in environmental health for more than a decade. Her work is focused on the implementation of the operational, analytical, and territorial, as well as transdisciplinary projects that address the impact and dynamics of urbanization, transformation in the use of soil, and climate change affecting human health. Specifically, she investigates health problems such as albeburosis, Chagas, and lepra. She also assesses the affectations of fire and in cities due to climate change. Mariana will present streaming. Let's give the floor to Mariana.
Hello, good afternoon. Can you hear me good? Please confirm. We hear you well, Mariana. I don't know if you can hear me, but Okay, I can hear you now. Great. We hear you as well, so you have 20 minutes for your presentation. Excellent. Thank you very much. Good afternoon to each of you. It is a pleasure to be and an honor to be here with you in the scientific conference in the uh, ISP. Thank you to the organizers, and I'm going to try to be very clear and concrete and respect time. I am Dr. Mariana Montecacosta, a doctor in biological science. I am from Argentina, from INS, and I am also one of the directors of one of the 14 Centers Institutes that is in the country. The idea today is to share with you a little bit about a project that we have been carrying out in the Institute together with a team that is interdisciplinary regarding the development of a monitoring system of the risk to the health due to forest fires. So, why do we talk about fires and health? Well, the researchers before I spoke have been commenting, and many of you as well must know. But for me, it's important to share with you the risk and the impact that is our South America is having in the region in a sustained way in the frequency of these fires. Currently, in February 2025, WHO has also given a very specific report that I recommend you can read, not only about the impact of burning, but also what diseases are the ones that they start to generate and the impact that it has not only in Argentina, but as well Brazil and all the other countries that are around us, that many of us live in these countries of the Southern Cone. And the other factors that contribute are highlighted, and I think that we have a consensus about the causes about these, and this has to do with the change of the use of the floor. We have extensive agriculture, for example. In my country, in Argentina, we have very strong agriculture, and we're also agro exporters. So we have the associated with the use of the floors, and the fires are affecting us. On the other hand, we also have the extreme climate conditions, which is where we are now in the form of climate change and health. These prolonged droughts and heat waves, and we also have different changes having to do with climate change, like, for example, the Nino. Going to Argentina specifically, I would like to tell you a little about that in Argentina, in my country, not only are we increasing in fires in my country and in the region, but also the increase of communication or mainstream media that visibilize this as a problem that can be seen not only in the municipalities and provinces, but also nationally. The Argentina, we are starting to see forest fires as a problem, and we are debating how it impacts us. And that is where we ask, what can we do from health points of view, not only to see the impact, but to see what strategies can we generate. And in this sense, in 2022, we generated from the national government and from the executive power a decree up till last year. And on the right hand side, you can see this is something that we elaborated by our team, where you can see the different municipalities that were impacted by the burning of the forest fire. So we start visibilizing not only the burn of biomass as an important problem to be evaluated from the environmental ministries and all of the environmental justice, but also everything that it means and how it affects. So how does this affect? How does the smoke affect And I mean smoke with everything that it means in the quality of the air, but all of its density, with all of the different components that affect the health of the populations, not only in the immediate circle, but also in a scale that, on a scope that reaches other populations. So, with the work team, we started carrying out and thinking about how to carry out a development of a monitoring system, especially because in Argentina, and if you allow me to talk about my country, but this is always thinking about other countries in the region as well, because this is a big problem that we do not have monitoring of the air quality. So that brings on a little bit of a deficiency of how to assess the air quality if we do not have a sustained monitoring system. So we can talk that there is great consensus that the risk on health can be assessed regarding these variables: the threat, exposition in the territory, vulnerability, and always considering the capability of adaptation or resilience. So this is where we started to work, and the threat it was included. And when I talk about vulnerability now, I also include the resilience of the territory. So thinking about the threat as an expression that is extraordinary about a natural phenomenon, a space time that is determined, we can in the taking in consideration in terms of its strength, mass, duration, distribution, and frequency. So we talk about the smoke. For example, you have the heat focus that many of you have seen this today and in other congresses as well, and obviously that is a very strong indicator.
¿Aló? Ahí sí. Ya.
Well,
Bueno, la verdad nunca nos hemos llevado bien con Argentina.
we never
Eso lo
got
sabemos.
along so well with Argentina. We knew that beforehand.
Eso lo sabemos.
We know that.
Entonces, ¿cuál es el
So
resultado?
that's the result.
¿Seguimos haciendo intentos para que
So,
nos
will
Argentina
we
o
continue
avanzamos
trying with Argentina
a
or
otra
should we go
posición
to
y
the
dejamos a
next
Mariana ahí quieta?
person and leave Mariana?
Podemos hacer el cambio. Entonces
We can
pasamos
do
con
the switch.
Ecuador.
So, let's go on with Ecuador then. So, let me find the presenter.
Era para demostrar que le tenía fe a Argentina, pero no.
Just to prove that I had faith that Argentina would be back in time, but
Ya,
there we go.
entonces
So,
continuamos
let's
con la
continue then with the
presentación
presentation
que se observa
that
aceleración acelerada
accelerated invasion of
bajo
Aedes
el cambio
aegypti under the
climático,
climate
el rol de
change,
la
the
técnica
role
in
of
situ en
sterile insect
Ecuador.
technique
Esta
in
estará
Ecuador.
desarrollada a cargo del
This will
doctor
be
Marco Sánchez
Dr.
del
Marco
Instituto
Sanchez
Nacional de
from the
Investigación de
National
Salud
Institute
Pública
of
de
Research
Ecuador.
of Public Health in
El doctor
Ecuador.
Sánchez es médico
Dr.
veterinario
Sanchez is a veterinarian
zootecnista,
doctor.
magíster
He has a
en
master's in
salud pública
epidemiology and public
veterinaria.
health in
Es
veterinarian.
responsable del Centro de
He's
Investigación y
responsible for
Exposición
research
del doctor
ISPI
Leopoldo
Institute and
Isquieta Pérez,
Leopoldo Isquieta
especialista en
Pérez. He's
modelos
specialist in
epidemiológicos socioculturales
epidemiological model and
aplicados a la salud humana, animal y ambiental. Por
focused
este
on
enfoque, él es
environment,
parte de la
animal, and
organización. También tiene un
human. He
máster en
also has a
ciencia
focus on
salud
research in
pública
public
mediante
health
análisis de datos,
through data
con softwares
analysis
de
with free
UNIS, Geodat,
software
RStudio,
and different
lenguaje de
studies and
programación
language of
para estudios
programming to
epidemiológicos
be
de
able to
salud
study
pública, tanto
epidemics in
sanitaria
public
como
health.
silvestre
He
y
also has
sociobiológica,
studies in
así
different
como
kinds
enfermedades
of
zoonóticas y
species
parasitarias.
and
Agradecemos
zoonotics and
entonces a
parasites.
Marco
So we would
quien
like to thank
dé su
Marco,
presentación.
who is going to now come and give us a presentation. I'm one of those who moves around a lot. So, first of all, I would like to thank you so much. It is a pleasure and an honor to be here with you, sharing a little bit. Yeah, to share a little bit about our experience. And I would like to thank the organizers. So I'm going to talk a little bit about the accelerated invasion of Aedes aegypti under the climate change, the role of the sterile insect technique. So to talk about this, I have prepared a little bit of a scheme that we are going to talk about. We'll have a general introduction, then we will talk about climate change and its effects, and then we will talk about the distribution of the altitudinal distribution of the Aedes aegypti in Ecuador and the evolution of cases of dengue in Ecuador and the role of the sterile insect technique in Ecuador and a few conclusions. First, then I'm going to talk about Aedes aegypti, which is a mosquito, a species that prospers in populated areas and is found in all of the tropical areas of the world. And this is the main vector of diseases, like, for example, dengue, Zika, chikungunya, and a different yellow fever, among others. And in a study, they determined that for Ecuador, there would be different types to see the genetic diversity of the populations of Egypti. So, to be able to talk a little bit about the climate change, we would be able to talk about the effect of the climate change can modify the temporal spaces of the vectors. I think I may have to come. We are getting a little bit of the microphone being cut off. 1 second, please. Okay, here we go. Now, so I was saying that the effect of climate change can modify the temporal spaces of the vectors, and it can promote the expansion of adequate habitat for these species. These species that were limited to certain regions could now survive and reproduce in more elevated areas where before that, because before they were colder, so that was not correct for them. So according to a study in Montenegro having to do with the global warming, they would describe that there was an increase from 2010 to 2040, there could be an increase of zero point 0.6 to 1.4 degrees. And for 2040 to 2070, an increase of 1.8 to 4.6 degrees. According to the ideal model of the habitat for dengue that is carried out in 2024, they determined that almost 55.5% of the territory of Ecuador could be exposed to middle or high risk for this mosquito, and this is having to do with the presence of the vector. And there were many different layers or conditions that affected, for example, the growth of the population, the recollection of trash, where they collect trash, the height, water provision, vegetation index, sewers, and rain. But the growth of the population is the main one, which was 63.3%. We can also mention studies like the one of Escobar in 2016 that predicted that there would be a decrease of dengue. This is from the study of Nature that talked about that for 2030 there would be a decrease in dengue in the presence of this vector. But we are 5 years away from 2030. We're getting up there, and that is not happening. We actually have an increase of cases. At least in Ecuador, that's the way it has been. What does coincide is that now it's going a little bit further north and towards the Andes, which can be seen. And in 2021, in a study of Colon Gonzalez, we can see that this factor is coincides, and you can see the different areas. And of course, in Ecuador, up to some years ago, we had dengue, we had this mosquito up to 2,000 meters approximately. And now, in the INSPE and the institute has now recollected species that are over that are 1,650 meters above sea level. So you can start seeing changes basically. So you can describe the geography of Ecuador as well. Well, Ecuador is has the equinox line, the Ecuador, that's why it has that name. And this means that it is in the tropical part of the world, and it has a climate that is that is warm and humid, and it is divided into four regions, like I indicated here. And it has the coast, Sierra, Vente, and Insular, or Galapagos Islands. So you have the coast that is also called the Amazon, and then you have in the coast, in the Amazon part, it has the wind from the Amazons, and they are retained by the um, Andes Mountains, making that it is very humid and that it has a lot of rain. However, the other side, as you can see here, the mountain range with the Andes, it has high altitude, and as you can see, that division is as a province. When I talk about regions and I describe four, it's not like here in Chile where you have many regions. You would say, well, in Ecuador there's only four regions. No, it is a division that, even though it is a political division, it also has to do with the geographical distribution. And in the coast and also the island and the Galapagos Islands, we have this influence from the Nino 3.4 and the El Nino 1 plus 2 coinciding with Peru on that. And you can also see here the graph on the anomalies of the temperature for Ecuador based. Uh, Here you can see from 1901 this graph was is not mine. I took it from Hopkins. And there in green, you can see from 1091 to 24, and you can see those climate going, those peaks, and you can see the cases of accumulated cases of dengue for Ecuador, and this is what we report, and you can see the peaks. And we can all see the peaks there, how they increase, and at the end, that long one, that high one, 2024, it coincides with what I was saying, that there is no decrease in dengue. And as you can see, if we cut this green line and we put it, you can see the coincidence of these peaks. So I have another graph here of the index of the cold and warm in the region 3.4 of El Nino together with the slide I was telling you about, and we can see that there's a certain pattern in the peaks of the temperature goes up and there is the growth in cases. So this is a small video about the evolution of dengue with the alarms in Ecuador, what we report when initially you see that the cases are centered on the coastal area, and you can start see the progression going up on the Andean area, and then it goes more to the Amazon in 2022. For the first time, you see the Galapagos Islands, and now, of course, we can see how, if we can remember from before, the Andes Mountains part, you have the provinces that we have from the Sierra start slowly getting in color as well. I had, um, it seems like the video is a little bit slow, but this is a projection for 20 years. This is a video that shows us a little bit of the growth for 30 years, and you can see that in 2024, how it all gets full of color. And it's important to highlight that this division that this was carried out with is by province. In other words, it is a political administrative division that is the largest one. So it corresponds. So I could say that maybe it would be more the regions, but we have divisions that are political and administrative divisions, like for example the cantons and uh, the chapels. They say that it is not churches. It is a small division, like a commune or a district. So if you see the cases, they're a little bit closer to the coast or to the east. So the technique of the what is the technique of the sterile insect? This is a method that is biological, and the idea is to release in a systematic way sterilized males that are sterilized, and that they can then be with the wild female so that they do not produce viable descendants. So they'll put the eggs, but they will not be viable. They are sterile. So the technique of the sterile insect is not new. It has been here around since the 40s with Edward and with another species, with the Coccomium minivorax. And this is a worm. The veterinarians probably know what I'm talking about. And since then, up till now, they have carried out there has been a lot of progress, and they have been able to massify and have the separation and the manipulation, the radiation, and the quality control, and all these other steps. And here you can see a map of where it is implemented around the world, this sterile insect technique. And based on a focus that is conditioned by phases, or PSA, where the volume of activity and investment will grow in the opposite. So you will not be able to go forward in a phase without completing the previous phase. So the pyramid shows the level that is necessary for each of the phases. And here you can see that on the map, phase 0 is not in there are certain places that are not there, but you have operational phase as well as 0, but there is only one that is in pre-operational phase, which is Brazil. So what happens in Ecuador? How is the progress of Ecuador? Well, the in they have 6 steps that I would say are transversal. They are described as 1 through 6. However, they are transversal and can be repeated, and they are necessary to continue doing the different steps. So as you can see here in the pyramid, the Ecuador, after these steps, we could say that we are in the pre-operational phase, phase 3, and looking for 2026 to be able to reach the operational phase. And to describe the steps quickly, the baseline is very important because that is where we have a evaluation of the area where it is going to be carried out, to have the collection, and we are going to establish the strain in laboratory. And we also have a characterization of the population. We have ecology and behavior studies. We have logistic of sterilization and transport, and we have protocol before and after so that this can all be maintained and can be feasible throughout the full process and the different phases. There's also a design of protocol of liberation and monitoring. And once this, we can talk about then of raising colonies, and this strain, you have an eclosion of the eggs, and then they raise these immature so that then we can have testing. I think there's a video. So we separate sexes of the at that stage to be able to go to the process 2 where they raise the adults and then they have a massive production of eggs, because that is what the objective is, to produce eggs. And then we are that way going to have the sterile male for irradiation, and that we have a hatching. And then we are going to have also different testing, and it's progressive, and we are going to be increasing the numbers of eggs, the sterile males for radiation, and then we will also have different and then we will prepare them for them to be sent out. So the male mosquitoes are sent to the irradiation places, so they have standard doses for radiation to guarantee total sterility, which is very important, and this is through different tests. And there is a determined number of males who are being radiated, and it's important that we can take into consideration that it will be necessary to change certain doses according to the number of specimens that are going to be radiated. And once they have received the radiation, the sterile males are sent to the field where they have an important phase of pre-adaptation of these mosquitoes. And this is key to remember. Sometimes you may not be seeing it here, but up till now, the mosquito has been many days since they come. They have radiation, and then to be transported, they have to be placed under temperatures that will avoid that they will be, I don't know if you know a mosquito, that they have the different parts of their body, so you have to have them completely well to be able to fly, because it's important that they can compete with the other male and be with the female mosquitoes in the field.
To release and the pre-adaptation of them, there is a we applied a fluorescent paint, and with it we can move to the next stage of marking, liberation, and capture. And this is important because the mosquitoes are marked, released, and then recaptured with the goal of assessing the state of them in the survey area and to assess what we are doing. And for a particular phase, we always do quality control in these field labs where we guarantee that the mosquito complies with the behavior, physical, and biological standards that are necessary for this natural environment so that they can compete with other males. And we do a lot of evaluations, and as you can see here, we have those squares where we do the testings, where we do the quality control of the mosquitoes. And there is a perpendicular phase here as well that's very important, which is uh, the relationship with the community. And it's important to empower the people so that they know what we are doing and so that they know that their support is necessary. And in which way? Well, support in different stages. For example, by not applying insecticides or by suppressing certain practices that are suitable for these mosquitoes. And these are a few images that show successful cases of these work in different countries, in countries such as Brazil, Spain, China, and Singapore. A few conclusions of this work and an overview of everything we've seen so far is that climate change can represent a determinant in the alteration of the populational dynamic of Aegypti, favoring their geographical expansion to either areas where there's higher altitude and warmer regions where there wasn't a presence of this vector in the past and now is there. And this geographic factor requires an adaptation with new vigilant surveillance strategies. As you can see, this work requires several critical components to effectively implement it, such as colonization and massive raising of these mosquitoes, all of it under controlled conditions with sterilization to precise doses of radiation that guarantee the fact that these are sterile mosquitoes and and allows these mosquitoes in turn to compete for mating. All of these should be made in the right density with a rigorous monitoring system to assess the impact on the wild population. So it is important to have a tool such as TIE and to have them validated in operation because it allows us to be more sustainable, efficient, and to act faster whenever we have outbreaks of dengue or arbovirus transmitted through Aedes aegypti. So basically, TIE can be a preventive measure when facing adaptation plans for climate change. Thank you very much, and thank you to everyone who made it possible for us to be here to work in climate change. And I would also like to quote those who are behind us and that make it happen and make it possible to do this work in Ecuador. Thank you, Dr. Margot Vidal, who was a mentor of the regional group of research and climate change and health. Thank you to my bosses in Ecuador that allowed me to be here, such as Dr. Pulara Borja, Dr. Andres Carrasco, and Dr. Varsovia Sevat, who is the mentor of TIE in Ecuador, along with Dr. Patricio Ponce, and they made it possible to do these wonderful collaborations and with work together with um, in addition to the work that Fabi from Bolivia mentioned and allowed us to support the work that they are doing in other areas. So thank you. Thank you, Marco, for the presentation. Thank you for the interesting presentation. I did not know about the technique of the sterile insect. It looks promising. However, I have a few questions. The first is that in your team, have you ever tried to assess the possible ecological implications or consequences that releasing these sterile insects might cause? Because, of course, there is a biological ensemble that will be affected by that. But my second question is also related to what you were doing because you're facing the vectors of the arbovirus. And I believe that in this case, these are from the Aedes gender and the affectation that it has to people. However, we're not doing our work directly with the virus. We know all mosquitoes are vectors and have arbovirus. So my question is, have you ever done any studies regarding the entropism of, of of these different ones, and what's the pressure that might be induced when you reduce the population of these mosquitoes because viruses evolve, and that might cause selective pressure, and we may not have a reduction of the virus, although we are reducing the amount of mosquitoes that carry this gender. But what happens with viral entropism? And that has happened and in other areas as well. Yes. So I started the presentation talking about Aedes aegypti is an anthropic species, plague. I believe this, although some studies say this isn't really like that, but they are not part of the atrophic chain. And I think this is a mosquito with anthropogenic preferences that are highly marked, and so I don't think it affects directly other ecosystems. I would say no. The other thing is that another thing that's very important is that it's the local strain and having this local strain. So we gather the local strain, we extract it from there, and we raise it in the laboratory. We're not introducing a strain from other areas. It's only from the area in which it will be released. And so I think I answered the first question. And the second question, it's more related to the virus, right? And I didn't explain this in depth, um, but right here we have the ecology of the disease that might be shared with Pictus, but in Ecuador we didn't have this since 2019, I think I'm not wrong, but it was the first time that we found Albopictus in the country, and we have seen these in 3 or 4 specific regions, and we've made some studies on this, and it's very specific for some areas. So if there was a competition, I think it would be with this other vector that has the same characteristics. I think that would answer the questions. The question. Thank you. Yes, a little. Thank you. Yeah, we can continue with the questions during dessert. Thank you, Marco, for your presentation. Here's a gift from the institution to you. Big round of applause for him. And if there are more questions, or please do it during the coffee break. Let's continue with Mariana's presentation. Is she online? We've always liked Argentinians. That's just a myth that we don't like Argentinians. Can you hear me? They were talking uh, about you very badly. I'm listening. I'm listening. You're blocking me. They say here that that's because you're Argentina, but that's absolutely not possible. Yeah, we will try not to win the next World Cup. Oh, I think the internet's bad again. I mean, we're losing you. Mariana, You have the floor. You can continue, and please summarize a bit of your presentation. I'll do everything I can. Thank you. All right. Although I have 20 minutes, I will do this very fast. I was talking about the development of a monitoring system of the risk of health due to forest, due to fires, and I started the presentation. I'm from Argentina. I am the director of one of the centers of research and health of Argentina. So, talking about fires as uh, an issue that has taken over the media, and we have this fire emergency, and the executive power at the national level puts in the agenda the multiple causes that are causing these fires. And in that sense, as a team, we ask ourselves how is affecting our health, this smoke, how is it affecting the health of our of Argentinians at the big scale? And we were able to try to understand this risk uh, through this algorithm that can be a little bit complex, but it's a way of thinking about trust based on the vulnerability. And I, you can look at the quote there and on how we discuss and why we chose these variables. So when my internet went down, I was just talking about this slide where we can assess smoke with the heat points, heat spots, and it's important to assess these uh, spots and to understand the intensity that they have. On the other hand, we have the chemical transportation model, and here are a few components that assess this value, and we also include here the main particles that are creating the biggest risk for human health. These help us as reference in Argentina, and now I'll share with you some of the names of these factors. The other thing is air quality, and air quality means that it was necessary for us to have indicators that help us understand how the concentration of these agents are in the air and to be able to assess them based on international standards and try to understand whether this is going to affect the population or not. Now, these standards can be um, discussed, but it is important to create a debate with them. So, our challenge in this project is to integrate this information to characterize the threat of this threat in the Argentinian territory. So, we started working uh, with all of these indirect mechanisms, and considering that Argentina has a deficit in the monitoring of air quality of the territories, and there's a specific monitoring, and because of that, we started creating these type of indirect assessments of the geospatial area. When I talk about these vulnerabilities, we talk about the conditions of the people and the people who inhabit these territories and the capacities that they have to anticipate to all of this. And we usually talk about resiliency when facing threats. And what we wanted to do when we were assessing vulnerability is not only looking at it from the biomedical perspective, and this means that it's not that we just wanted to identify groups with higher physiological susceptibilities, such as children, the elderly, or people with chronic disabilities. We also wanted to understand how the contribution of social sciences can include structural challenges. And when I talk about these social, economical, or structural challenges, I am talking about those factors that create an inequality compared with other uh, population. So we worked not only on that economic aspect, but also cultural aspect. That's what we mean when we talk about vulnerability. And when we talk about the biomedical aspect, we ask ourselves how the air contamination affects our health. And Fabiola already talked about this and mentioned the issue of fires and how it linked with the fires, and the PAHO already established what are the main risks affecting people's health in the entire region. And in that sense, we included these chronic disabilities to our model, to our analysis. And on the other hand, as I mentioned, the area of vulnerability, we started including it as an index of vulnerability that was created with the help of the Geographical Institute of Argentina. And it included all kinds of vulnerability in the face of disaster risks. These disaster risks can include many more disasters other than fires, and that's what we are working on at this moment because we want to create a very customized indicator to understand all of this. And the dimensions and variables that we are working with when working with this indicator are three. First, we have social conditions, education, health, and demography. Then we have habitation, housing conditions, the housing, basic service, access to that, and economic conditions, the work, education, and family. So we want to understand the perception of the risk that these people have. And we started to delve into surveys that will allow us to understand how the people who work and live in this territory perceive fires as a risk for their health, and not just from the biomedical point of view, but how do they perceive it? And that is a cultural dimension that allows us to see and understand risks differently than other communities. And so we have this challenge. On one hand, we have the threat, on the other, vulnerability with the social economical indicators, with the surveys that we use in order to have a risk map that will allow us to have almost immediate information received at least on a daily basis so that decision makers in Argentina can take these recommendations or give recommendations to the population in these cases. This project it started in 2023 with different institutions such as um, this, the National Center for Diagnosis and Research, the National Institute of Respiratory Diseases, the Informatics Department of Malbran, and then the High Special Studies Institute of the University of Córdoba. And something that I think it was significant to highlight is that something as complex as flow charts, heat charts, need need to have work together with other professionals as an axis, and it needs to be transdisciplinary. In order to work on this, we decided to work with professionals from different areas, and you can see them on the screen. We have professionals with degrees in biological science, biological chemistry, computation, environmental sciences, sociology, etc. Our study area, although Argentina is a very broad territory, we thought, how can we start working with our modeling and analysis? And we have one in the El Plata basin, and we chose it because it's the second basin, hydrographic basin after the Amazonas with sweet water, and because of the fires and the, the information that we had, and the location with the other countries around it, near that same basin, ecological rivers, geopolitical significance, and we marked those initial uh, as points where we focus our study in this La Plata Basin.
The basin is extensive. We would like to have a smaller one, so we have the Delta of Paraná. As you can see, this is in Argentina in front of our brother country, Paraguay, which is basically a sample of all of the basin of the Plata in total when it has to do with the crops, the territorial, and other areas. And in 2024, there was a study carried out where they evaluate the increase due to the hospitalization, due to the different strokes or uh, heart attacks and that have happened in the last few years. So you can see a little bit of the results. And from the initial that we started to have, we started to see the heat focus or the heat points. You can see the red points have all of the heat points that started to be registered and the hot spots that could be seen in all of that area. And then you can also see in a dynamic way that here you have the uh, points that have been burned and the points that where there is heat. So looking at this, we can start to see where the people were, who were the people that were the most affected. So we chose the population that is most exposed, has high dependency of the natural and environment, that has most vulnerability in these territories, and also with a focus on environmental justice that these fires bring in these territories. So there you have some of the names of some of the locations that we started to choose to start working on this study of fires and the impact on health. As I said, this also has to do with the territorial space when we start to see how basic needs that were not satisfied were marked, and we start to mark on the maps, having to see the risk index and the hot spots that we marked previously, and the basic needs that have not been satisfied. And you can see here on the left how gradually it goes from 0 to 100, right? In the center and in the south are the areas that are the ones who have 100% of the basic needs not satisfied. So what happens at this point? We start to develop a study through surveys or polls, because we consider that the cultural and perception part has basic information that complements the work that we were dealing with or we were handling. So we had 1,000 people who were surveyed in the territory of this delta, and there you can see that every circle or every pie talks about one location that was surveyed, and each color talks about the perception to any exposition depending on the person who has been surveyed. Look at the red and the yellow. They are all more in the south and the north as well in the responses. So this means that not only did we perceive risk in the indicators and in the heat focus, but they could also visualize these fires, and you can see the risk. But the risk that they had was more normalized. Fires was so normal in their day to day that they did not see it as something so high risk within their lives. And what I just said is not something out of normal in health sectors. Many times we have other diseases that are affect certain territories. However, it attacks so strongly to certain populations, and the populations end up normalizing these issues. What other data do we have? Look here, everything that is light blue, you can see yellow is the area being studied. And here we could see the heat focus, the heat points, and you could see the geospace to see how they are connected with their perception. As I said previously, there is an association between what people perceived and the heat focus. However, the risk was very different regarding normalization of a fire in their territory. And closing, in case there's any questions, where are we now in all of this development of this project? First of all, having the installed capability in Argentina of a development of a model based on chronic exposition that combines air quality and populational vulnerability. On the other hand, we have the capability to have circulation per hour that can be modeled. You can see on the right there is a map that was carried out with the team where you can see the different heat focus and the index of air quality. Where in 24 hours in the national territory, you can see those having to do with the fire, how what the risk is in our country at that point. That way, as you can see, this is for 24 hours, but we could do it by hour. It generates the capability for our country. And we are also in the generation where we are developing a fire risk map with the potential to be able to give information to the decision makers. And as challenge, what we want to do is go deeper with this integration of data. Not only that it can have biomedical data, but also it can be territorial data, data connected with the most affected people, and that it can also have connection with the decision makers to validate in a territorial way. So this means an adaptation with the local situations and to be able to go, we are planning to move to the territory to generate interaction and a connection of the progress of this project with those who are affected, to be able to incorporate this information and send these index to the territory, and that way amplify the key motors that can give us this information and how they can make decisions for themselves or for their communities to give a response to these fires. And lastly but not least, also financing that we need for a development like this, alliances with different institutions, and this also puts in evidence, and I want to propose the alliance with all of the institutes that are listening to us to be able to generate development not only in Argentina but also in the southern cone. In this sense, I would like to place, we just delivered information and we want to generate information. We have a magazine or a journal, which is also work that we have been able to share, having information about what we are carrying out, and allows us to have alliances with other institutes of the country as well as the whole region. So I would like to thank you all. I place not only my name and my link and email for you to connect, but I also have a QR of our institution so you can connect. Thank you very much. Thank you very much, Mariana, for your presentation. I don't know if you can hear me okay. Yes. I don't know if anybody has a question for Mariana. Yes, there is. One moment, the microphone. Hello, Mariana. Thank you for the presentation. I wanted to ask you, why are fires being measured only on the impact of the health only by the smoke? Because the fire can also impact the health through other mechanisms like stress, fear, population having to move. Are those also integrated in the models at this time? No. Okay. No, I'm sorry, I didn't hear your name. Valentina. Thank you for your question. It is very interesting what you are bringing up. The reality is, I'm sure you have knowledge of everything that the fire affects. All of those different areas for us are a great challenge to be able to include everything that it affects, and not only this part. As you say, it also has mental health issues. For us, it is key to think also that from the modeled part, it's a little bit more difficult to be able to have access to that information. However, we are not leaving it. We're trying to see how we can include it, whether it be through polls or different areas. And in our mind, we would like to do that as second step, and these can be indicators, even though it's a little bit more difficult to include them in a modeling that is more chemical, and it has to do with the concentration of agents, and it has to do, as I said at the beginning, in Argentina, there is no surveillance on this site to be able to develop it. So I think that what you are saying at this time we are not carrying out because this was preliminary, but we would like to include all of the different areas and inform how the fires are affecting different areas of health. Thank you very much.
Gracias, Mariana, por tu
Thank you,
presentación.
Mariana, for your
Muy
presentation. We are
importante que
about
la producción va a tener
finished with
que
time,
buscar
but you're
energía
going to have to
renovable.
travel to Chile to come get your gift that we have for you.
No le
So
vamos a
don't, we're
enviar
not going to
no le
send it to
vamos a
you,
enviar
so
esto gratis. Va a tener que
because you're going to have to
buscarlo.
come get it yourself.
Muchas gracias, Mariana. Muchas gracias. Le damos un aplauso a Mariana por su
A warm
presentación.
applause to Mariana for her presentation.
Antes del último
Before the last
póster
coffee that we
y
have and the last
de póster, ya vamos
poster
a
evaluation that we have
dar la
programmed for
bienvenida.
today at
Vamos a
4:30,
pasar
we are
con
going to go
la
forward
doctora Margot Vidal
with
del
Dr.
Instituto
Margot
Nacional de Salud de
Vidal
Perú.
from the
Ella nos va a
NHI from Peru.
hacer
She's going to share
resultados
with us the first two
de
regional forms of the
esta clínica la
NIHIs of
doctora
South
Vidal
America. Dr.
es
Vidal
magíster en
is
epidemiología
surgeon,
egresada de la Universidad
master's
Peruana
in
Cayetano Heredia,
epidemiology, and she
presidenta
is
del
also
equipo
president of the technical
interdisciplinario de
team
la
that is
Nacional o del
multidisciplinary
Instituto Nacional de
of the
Salud de Perú.
National
Es
Health Institute of
subdirectora de
Peru.
la
She's ex
subdirección de
subdirector of
nutrición y
subdirection
laboratorio
of
de la
research and
Organización Mundial
non-transmittable
y es
diseases.
ejecutiva de la
She is
Conferencia
executive
Sudamericana Internacional
director
de
in the
Epidemiología y Prevención
epidemiology,
de
prevention, and
Enfermedades en Perú.
disease
Es
control in Peru, ex
subgerente de servicios
sub-manager
finales en la
of
subdirección
final services, which is called a
egresada
salud.
del programa de alta gerencia del
She
Instituto
has
Nacional
been
de
in the
Prevención
national
y Control de Enfermedades en
public
Perú.
health
Diplomada en
management.
analítico de datos en epidemiología clínica,
She also has
egresada del
clinical
Instituto Nacional de Posgrado
epidemiological
en
studies
Ética de la
and
Investigación
also ethics of
psicosocial
biomedic
de la
studies
Universidad de Chile
and
y
psychosocial
becaria también
studies,
del Fogarte
and
Internacional
also from the
de
Fogart
USA.
International from the United States.
Docente del
She's a
postdoctorado
teacher
en la
of pre and
Universidad
post
Católica de La Calera. Dejo
um,
con ustedes a la doctora Margot Vidal.
graduate, and so I will leave with you now, Dr. Margot. I would like to greet all of you. First of all, I would like to thank the organization of this event for having made this possible for us to come, to be able to have representatives of the different NHI from South America and to be in this forum. I would like to talk about a small modification. I'm going to talk about the result of the three forums, including the one that we had yesterday in the afternoon as well. So to begin, I would like to comment how it is that this idea came up of uniting the NHI of South America. It started by asking what is the role that we had within climate change. So basically, we went to the mission that we have in the NHI in Peru, which is very similar to the ones, the rest of the ones of Latin America and even probably around the world, which has to do with a lot of research. And this role places us in the responsibility of leading research in climate change and health. So the first thing that I did was a quick search about the different research that exists in the world and compare them to climate change and health and other diseases. And if you can see quickly, the proportion is minimal. We have climate change, there is something, but when we go to climate change and health, it practically disappears. So the proportion that we have of public studies around those two things is not enough to be able to talk about it in an informed way. So that is why we wanted to unite all of the national health institutes because we have common problems. And that is how we had our first forum of NHIs in South America. And it was carried out in the first Congress of the first International Research Climate Change and Health. That was a conference that was carried out in Peru in 2023. This conference, similar to what we do in climate change and health, was carried out with no budget because even though there is a budget in climate change, it has to do more with the environmental issues. But when we want to unite it with health, the budget disappears. So we decided to carry out the first Congress to be able to make visible what there already was having to do with research, because even though it was very little, there still was some, even though maybe it hadn't been shared enough. So thanks to the support of many different institutions in my country that united to our dream, and we have Horas CONU and the Ministry of Education, the PHO, the International Migration Organization, and many others. We united to be able to make this research on climate change and health together, and it helped us bring of the different people from the NHI, basically as what we are doing now. So that is the picture of the first group. You have seen those who have been speaking before me. You can see that there are many of the same ones since the same day. And what is amazing is that if you see the list, is that most of the countries of South America participated in this first forum. So we carried out a vote, and with unanimous voting, we started to elaborate the agenda for research, not of each country, but of all of South America, identifying once again, as I commented, problems that we have in common. So we have also continued and we went to the second forum. The second forum was carried out in the second research congress of climate change and health, which was carried out in Mexico last year. And this second Congress, we propose from the second forum to form the research network on climate change and health in South America. We also presented the progress that we had in the agenda that we had initially agreed on to work on. So here I place the names again of all of those who have been working in the second forum. Many are repeated, however, some have been changed because each year there are political changes that sometimes cause some representatives to change. But I think it is great to highlight that once again all of the South American countries are represented in the second forum. As I said, we also had the network, and we wanted to show what we had the progress we had had in the research agenda. So what I'm going to show and come in detail now about what we have been able to have progress on having to do with the agenda. We have gone 4 stages. The first one was to identify and prioritize the health problems due to climate change that were common in the region. The second one, that each one of the problems that existed, we wanted to elaborate strategic objectives, and for each objective, we wanted to identify needs for research that then are qualified and prioritized, because as you know, we cannot cover all problems and all research needs. We always have to prioritize. And in my country, at least, the budget that is not, we do not have too much budget, so we have to prioritize. With all of these, so all of this together is what gives us the agenda of what we are going to research. So what we have gone forward is the first stage. The first stage is identification and prioritization of the health issues. So there was a list that was elaborated a preliminary way, and it was then filled in by the different representatives of the NHI, and we qualified these problems. We grouped some of them, and we finally voted for which problems we will be able to work on. And quickly, I'm going to show you this. We elaborated the first list of problems based on 2 reports. The problems are by the PHO and the Lancet Countdown and the climate change in South America. And there was also a list to be able to determine that then it was completed by the NHI groups. We made a longer list, and the last 3 are the ones that were incorporated. And then we qualified by 3 criteria, which was the knowledge gap, the fact to be able to intervene, and the consequences, and we did the same for each problem. We voted in a virtual way because the last forum was virtual, was online, and consolidating the score, we had the order of the problems, and then we voted for how many problems we could work on, being realistic. And we ended up saying that we are going to talk about work on 5 programs. And here's the results by the order. And we have here. I'm sure that this is not strange because Mariana commented on this quite a bit, and also Fabiola from Bolivia, the diseases of mental health and psychosocial ones, and also those non-transmittable diseases, food safety, and those that are through water and vegetables.
This is the working group of the second forum. It was virtual because we couldn't attend the second congress. This did not stop our work. And now I want to tell you about what we did yesterday. Yesterday we gathered in a parallel room from 2 p.m. to 6 p.m. and we couldn't conclude the session and everybody was tired, but there was a really beautiful discussion about where are we heading to now. And what was the first thing that we proposed in the beginning of the first forum was to increase from 5 problems to 6 problems. And the fifth problem included 2 problems: diseases transmitted by water and by vectors. However, these two are things that are completely different, and when we put them in the same problem, it was very complicated to address it. And we decided to address 6 problems. In the end, it was the same, but we decided to separate it. And we can confirm that it will be 6 problems from now on, but apparently the acceptance is positive. Now, something that I mentioned yesterday during this stage, which is the need of having research, is that there's a group of key actors. And I think that it's important for you to listen to this because this backs what we are doing. And there is a group there, which are the international experts in climate change and health. And in this case, I gave you a few names such as PAHO, Andean Community, Mercosur. Several have done uh, have made reports and research on this, and they are part of this commission of research. We also have regional researchers. One part of them is INS, which are the deans of investigations and, let's say, experts in methodology, those who work in national health institutes, but nobody's an expert. And then we have researchers who are not part of these institutes, and so maybe any scientists who wants to be part of this or part of this group, and they should be part of this group. And something else that we believe it's very significant as well is decision makers. So what's the use of having an agenda, put a list, make this list, if decision makers are not betting on working on this agenda? So we have these 3 key stakeholders that have to agree on things and together decide which priorities of research we are going to have. The third forum was only focused on the first problem, the one problem that had the highest scores. After talking about this problem, we are working on this problem, and this is the first one, which is health problems due to forest fires. There was a huge discussion about this, and this is a photo that won an award that we did at the National Institute in Peru. This was a contest for the population to share with us through a picture what's the impact of climate change in their location, and from that photo they should share what's the affectation to their health. And no surprise, it was forest fires. That was the winning picture. This picture is of an area of my country in Puno, of an indigenous community, and the title of the person who shared the photo is called Aymara Women After suppressing a forest fire near their houses. And I'll share with you a little bit about the context of what she shared with us. And it says, in the last couple of years, forest fires have increased in the entire world, and in Peru have caused big losses of major losses of vegetation and weeds, such as the loss of human lives. And that is because in these remote areas where it's hard to access, the help to suppress fires takes longer to get there or it never gets there. And I think that's her perspective. she's there in the picture after suppressing the fire on her own. I imagine it wasn't such a big fire because they were able to do it, but the people who live in these areas face these fires year after year with scarce resources and without the necessary help. The inhabitants are affected by because of they are breathing the smokes with contaminants. And in addition to sending the picture and the context, we asked them to give us an idea of research, and the population isn't used to sending these ideas of research. And what we included in the text there, where it says idea of research, something that we um, fixed because they are not used to sending this type of information. But this helped us realize the issues we have in certain areas in our country that isn't really part of the problems of the nation. So it's important that whenever we have or establish priorities at the national level, we tend to forget what happens at every region. And it is important for every region to do the same exercise because it's very likely that they will have different issues. Okay, this is a context for what we did and to share with you why we started working with forest fires. So yesterday's program started with a methodology that we wanted to apply for the agenda of forest fires. And then we had several lectures of international experts that were presenting so that we could have a reference of the results of these reports. Because as I said, we are not experts in forest fires. After the presentations of these reports, all of them were very interesting, including the one of Sandra Contes. You know her. She presented this morning as well. She talked about the situation of the health uh, that are facing the forest fires in South America. After that, we what we did was identifying the main issues caused by forest fires. And then we had to draft the strategic goals. And what I'm showing you here is a list of the problems because we had several problems, health problems, and somebody asked this question, but we really do have multiple aspects that can be affected by forest fires in terms of health. So what we did was just making a list of all of them, and what we finally did was drafting the strategic goals in a very simple slide. But doing so took us for at least an hour to agree on what could be the goals, which are the pillars in order to identify the needs of research based on each one of these strategic goals. And this is an something that will be fixed. It can change, but it is important to me, and I don't think we should move too farther away from this because in the end, authorities and researchers need to accept the work that we are doing here. So we are taking baby steps in order for this work to be validated by every stakeholder. All right, so this is a photo of the participation. It was mixed. Several were here, and the rest were participating online. This is a picture uh, of some of them, the people who worked here, and then also you can see in the picture those who participated remotely from Uruguay, Brazil, and at the regional level. As you can see, we have the Latin American Center for Change, for Climate Change and Health, and we have the Latin Network of Climate and Health that participated as well, and Dr. Bengolea, who is the coordinator of the Andean Network of Health Institutes, RICE. RAIS. And what we are doing is including several groups so that they can identify the needs of research to uh, create these agendas so that all stakeholders can participate and make decisions. And that's all from the first part of my presentation. I believe that we have a break now, and after that, I will continue because what I will say is what's to come after this. Esteban? Thank you, everyone. They tell me I can continue with the second part, and it's smaller, so let's do it. Do you have the presentation? All right, the second part of the presentation is very brief, don't worry. So what I will be addressing is what's to come for the next forums. I share with you that we worked on 4 stages. We are currently at the first stage. We have already identified the problems. We established it's 6 problems instead of 5, and we're focusing only on the first problem and the second stage of it, which is elaborating strategic goals. The problem that we discussed yesterday is this first problem, but we also have to work on the rest. Then identifying the key aspects of research and then the research priorities. That's what we need to do for forest fires. And then we have to complete the 4 stages for the rest of the problems of the list. And that's what we are missing and that we need to do. And when we have the agenda of the first problem ready, and something that's very necessary and important is to validate the results. We need to validate these results because each one of us, we need to do this, each one of us in each one of our countries, because so far the meetings have been macro between countries of South America, but we need each one of us in our countries to validate the results, which means, for example, in Chile, Chile, and just Chile, will have to validate the results that we are obtaining, and everyone can participate like that doing the same exercise. So in addition to this validation, we have to do a validation with other sectors because like many of the issues that we have in the health sector, this cannot be solved only from the health sector. We need the help of the Ministry of Agriculture, the Ministry of Education, like our colleague from Bolivia mentioned, Fabiola. Fires sometimes are produced due to cultural factors when they burn the land. So we need to educate the people from that perspective. This is a very participatory process, and we are trying to integrate every possible actor so that this can finally be accepted and so that we can be certain about the next steps for the future. The other thing that I mentioned is to continue doing the same work with the rest of the problems that we have listed. And something else that is for the after the break is we want to talk about the network of research of climate change and health for Latin America and the Caribbean, which is a confluence of 2 networks, the network of South America and the network of Central America and Mexico. And that is all. See, it was short. Thank you. Thank you, Margot. Excellent presentation and work of this network. Thank you so much for your effort. I want to hand you a gift from the Public Health Institute and Scientific Conference. We want to give this gift to you and a certificate of participation and acknowledgement of the activity of the scientific conference of the Institute. Thank you very much. Esteban, can you come? Because after that, he starts complaining that he's not in the picture.
Lo que me acaban de comunicar, por eso estaba yo allá.
I was over there because they were telling me something. It's not that I was just not doing anything. Let's move on to the posters. Coffee's a little late, later than we are. It's not our fault. So let's take a look at the posters for 10-15 minutes, and then they'll call us to go enjoy the coffee break. And we're also waiting for the rest of the people who are working in parallel with us. So go ahead, play some music. Thank you. let's resume today's program. The roundtables are it's going to be suspended for the day, so thank you everyone for your participation in our blog and on this program. Or this is our goodbye. Thank you. We'll see each other tomorrow once again, which is our last day of the scientific conference. We have presentations from the surveillance team and just surveillance team. Tomorrow we have presentations of the posters, so you're all welcome for tomorrow and last conference at 9 a.m. Thank you.